Good morning, everyone, and welcome to the Mid-Atlantic 4R Nutrient Stewardship 4R Symposium. My name is Lindsay Thompson, and I am the chair of the Delaware, Maryland 4R Alliance, and also a board member for Mid-Atlantic 4R Nutrient Stewardship Association. We're excited to have you all here today, and thank you so much for spending your time here learning about these important topics. I just wanted to, for those of you who are not aware of the 4R Alliances and the association and what we do, just give you a really brief overview. So the Pennsylvania 4R Alliance was actually started back in 2012, and then the Delaware Maryland 4R Alliance was started in 2015. And these are primary partnerships led by industry primarily, so the agribusinesses, many of which are present here today, but also the Nature Conservancy, one of the founding partners specifically for the Delaware Maryland 4R Alliance. And when we first started, our goal was that every acre of agricultural production in Pennsylvania, Delaware, and Maryland were going to be done according to the 4R principles. So the right source, rate, time, and place of nutrients. And the 4Rs are different for every acre, right? So just ensuring that every farmer and agribusiness professional had the information that they needed in order to make the best informed decisions for their individual farms. And since that time, we have really expanded in our partnerships. We've been the recipient of several different National Fish and Wildlife Innovative Nutrient and Sediment Reduction grants that we have used for education, training, and direct cost share to our farmers and agribusinesses to collect information on practices on the ground. We have conducted a barrier survey with the University of Delaware to better understand the barriers that farmers experience for our nutrient stewardship implementation. And we have also been the recipient of the Keith Campbell Foundation grants that have assisted us with our voucher program, as well as the development of many of the materials that you will see out there on our table. And so one of our annual events is the event that you are at right here today. So this started out as a field day, which we still think the field days are very important, but as a result of our recent strategic planning process, we determined that the way that the 4R alliances could be most impactful was to really focus on the trusted advisors. And those trusted advisors we found through our research are the agribusiness professionals, the nutrient management planners, and university extension that farmers go to for information. And so by training those professionals, we can then reach all of the farmers that they are working with as well. And so that brought us to the 4R Symposium that you're here today. We're really excited about our lineup here today. So as you can see up on the agenda, we have Dr. James White, who's going to talk to us about biologicals and biostimulants. Dr. Josh McGrath, who's going to talk to us about the FIRST tool. And then we have a very exciting farmer panel and Dr. Jared Miller talking to us about some grid soil sampling for variable rate application. And Dr. Nicole Fiorellino talking to us about high management wheat nutrient recommendations. And then last, but certainly not least, I would like to direct you to our sponsors that you saw looping on the presentation earlier. Without their support, none of this today would be possible. So any questions before we go ahead and get started with our first presentation? All right, hearing none, I'm going to turn it over to Dr. James White to talk to us about how plants use microbes to enhance development and acquire nutrients.
All right, so I'm happy to be here to be able to speak with you. What what I look at and what I'm going to talk about are how plants use microbes and why the biostimulant microbes actually work and how they work inside the plant. Also, this this relates to how plants use soil microbes. So how plants naturally, if you're growing plants organically, why you need healthy microbial rich soils, because plants actually need those microbes for their development and for their nutrition. And I'll explain how that. I'll show you how that works. Okay, so the microbes I'm talking about eventually become endophytes. Okay, endophytes are essentially non-pathogenic microbes that go in plants, that plants have inside them. And you may be familiar with the tall fescue, perennial ryegrass endophytes. Those are actually fungal endophytes. And this shows below one of the from tall fescue, I think. And you can see the hypha of the fungus inside the plant. Okay, so there are fungal endophytes, but there are also bacterial endophytes. All plants, including those grasses that have the fungal endophytes, they also have bacterial endophytes. And this is actually some microbes that we isolated from corn. It's something called micrococcus, but it makes these little tetrads, little clusters of bacteria. What you're seeing here, by the way, is a root tip cells of a rumex, a common weed, where we remove the microbes and we put that micrococcus in it. And now you can see in the cells of that root tip, you can see the bacteria that went in, and these are the tetrads that are inside the plant. You see they're actually in the cells. They're intracellular, so the microbes get absorbed into the cells of the root from the soil, from the outside. So plants are doing this. They're okay, and uh, why are they doing this? Why are they absorbing those microbes from the soils into the plants? Well, where they actually absorb them into are into the meristem. So the root tip meristem, they get them. Also. They cultivate them in the other meristems of the plant. They'll also cultivate microbes into the shoot meristem. So if you look at these meristems in almost any plant, you'll see, and those are leaf meristems, those are shoot meristems, those are root meristems. You'll see they're loaded with bacteria. They're absorbing bacteria from the soil and they're moving it throughout the plant and keeping them in these meristems. But why do they have them? They have them because they need those microbes for development. Those microbes produce hormones that the plant can detect and that the plant uses to grow. And I'll show you how that works. The also they are using the microbes, endophytic microbes, soil microbes that become endophytic. They're using them as a source of nutrients, and I'll explain how that works. And the okay, so what are the microbes? They're all that plants are getting. They're for the most part they're soil microbes, and plants just take them out of the soil. But they also adapt the microbes to the plant so that they then put them into the seeds. They go into the seeds or seed vectored, and they go all throughout the plant, providing nutrients in those meristems, right? And so they plant cultivates these microbes in the meristems, and I'll explain that. An example here is the rhizophagy process. Rhizophagy, also called the rhizophagy cycle. Rhizo for root, phagy for eating, and this was a process. I'll explain it, but it's a process whereby plants attract microbes to the root tips using exudates, absorb them, get nutrients from them, put the surviving microbes out back into the soil in root hairs, and this process, what I'm listing here are some papers that we did on this, and these are all published since 2018. So our first publication on rhizophagy cycle was probably in 2017, but this one is a complete story, and it's in the Microorganisms Journal. We had another one in Microorganisms Journal here that describes it a little more, and then we had some more studies here that was published in 2022. So those are papers that you can get to, and they're all open access online. We did not discover rhizophagy. It was discovered by scientists in Australia. We just worked on it and explained how it works. But these are some of these Queensland, Australian investigators. And this is from a paper that they did. The paper actually was called Turning the Table: Plants Consume Microbes as a Source of Nutrients. This was published also open access in PLOS One, so you can get all of these articles freely. There's no paywall. You can just get them. They're there free. You can download them and study them for yourself. These authors named it rhizophagy. Okay, so I should say what they show. This is from their paper, and if you look at E, you can see a root hair there. And in that, you see these green glowing little dots. Those are yeasts that they labeled with green fluorescent protein. Okay, so they could mark it, tag it, right, and track it. Tag it and track it. That's what they did. They tagged it and tracked it. And so wherever you see the green, that's where the microbe went in. So they tracked it in the plant. And then over a period of time, these microbes disappeared. So they concluded that they must be eating somehow, consuming these microbes. They named it rhizophagy, like I said, root phagy, root eating. So they get the credit for this, really much of this discovery. It turns out bacteria are involved in rhizophagy. Yeasts are involved in rhizophagy. That is, plants are absorbing these microbes from the soil. But we've recently found over the past year or two that also algae can be absorbed into the plant. And algae could provide nutrients, but algae could also carry other microbes, other bacterial microbes. Algae will have their own endophytes, so they'll carry those into plants. What you see below are images that show the bacteria now in plants over here in the root hair. And you can see that it's stained, so you can see the bacteria. If you don't have a stain, you can't see this. Okay, here, this shows a yeast in a root hair. See the yeasts coming out. They're actually, when they're inside, they're in a form of protoplasts. They don't have cell walls, and I'll explain that. They're just naked protoplasts floating around in the cytoplasm of those root cells. And you can see the algae here. And you can see an alga in the middle, losing its chlorophyll, and then you see bacteria on either side. So, okay, what we have shown is that in fact this is a cycle, and that plants are using exudates here at the root tip. They're attracting microbes to the tip. When those microbes come, they absorb those microbes into the cells at the tip. And then once they're inside, then the plant will hit those microbes with reactive oxygen, superoxide. And I'll explain that again, how it is. It's a simple process. Basically, plants are oxidizing these microbes. They oxidize the walls and the nutrients out of the microbes. Surviving microbes are in the form of a protoplast. Okay, so they don't have cell walls there. They're just like naked. Normally fungi and bacteria have a cell wall on them that protects them. Well, that's all gone. And so they just have their like membranes there in which they are floating around inside these plant cells, inside the cells of the plant. They will go into to the root cells, and then these root cells will differentiate a little bump on them that is essentially a root hair initial. That little bump turns into the root hair. And if there's bacteria that have been absorbed, then those root hair initials will fill up with bacteria, with these bacterial protoplasts. And then in those root hair initials, in the tip of those root hair initials, then what you have is those microbes will secrete hormones, growth hormones, ethylene and nitric oxide. Those are the two that we can detect. So it secretes those, and those hormones result in growth of the hairs. And so with the microbes there in those root cells, then they can form root hairs. If there are no microbes there, they cannot form root hairs. There's no root hair development without microbes in those root cells. Okay, if you look here at the image, you can see where the root hairs over here, but look at the tips. Those are bacteria that came out of those root hairs. The way the hairs work is they actually are microbe processing structures, and they will, as they grow, they eject microbes back into the soil. So the plant is repopulating the soil with these microbes that work with it. And those little bumps there, those are masses of bacteria that are coming out. I'll explain that process better. So it's a cycle, a rhizophagy cycle in which plants attract and absorb, get nutrients from microbes, and then put them back out, the survivors back out in the soil to get more nutrients. This actually shows a grass root uh, here, and you can see the tip here. That's where the microbes are going in, and you can see microbes there, around the tip. That's where the plant is putting exudates and cultivating and attracting those microbes. And here the microbes will come out of the tips. You don't see them in that image, but if you look here, you can see them here in this better magnification. Here you see the root hairs, and you can see the green dots there. Those are bacteria. This is fluorescent stain so that we can visualize those bacteria. And they come out every time there's growth in a hair. The bacteria come out of the, I should say, they don't come out, they're ejected by the plant. So this whole rhizophagy is under plant control. This shows a root. This is a grass, one of our invasive grasses that we study. And you can see the root here, branch root here, and you can see where the exudate zone is. You can see all these blue dots. Those are bacteria that this plant is cultivating at its root tip. And it's taken those bacteria into the cells right there at the root tip. You wouldn't be able to see any of those bacteria without a stain, at least not when they're inside. But here's an example. Here we use the stain crystal violet. And this is a root tip, and you can see where the bacteria go in. You can see they're rounded there where they went in this case. They're rounded. You look below, you can see those little round dots. But over here, this is the youngest part to the right. To the left is older. You can see what happens. Once you get past this entry area, this entry zone where the bacteria enter the root, then it hits the microbes with superoxide. And what you see here, this is stained with crystal violet, which stains the cell walls of the bacteria. You can see, if I look below, you can see how irregular this is. This is because the cell walls are being oxidized off of the bacteria. And as it turns out, so the cell walls that get deposited inside those plant cells, it turns out those cell walls have, there's a close-up of it. You can see the cell walls, the dark color, that's the cell wall coming off. The little light pink, those are the protoplasts. Those are the cells without cell walls. Okay, so that crystal violet stains the cell wall so we can see them coming off. And this is a common bacillus. This one actually is a biostimulant microbe that we got from, in this case, to play with this microbe, we got it from a company that they were using it. Well, this losing cell walls, oxidizing cell walls off of microbes gives the plant some whatever was on those cell walls, and they have in there some protein and phosphorus and metals, divalent metals, a lot of metals. When these bacteria were in soil, they accumulate these metals on their surface. And here you can see a bacteria. I mean, you can see these little white dots. Those are the metals that are on those bacteria. So plants can get these divalent metals, these nutrients, these micronutrients, or they're not all micronutrients. Calcium is one of them. They can get these by absorbing these microbes from the soil. And okay, so this actually shows, this is another plant, shows epidermal cells of the plant, but this is showing these protoplasts. And you can see the protoplasts inside. You can see there's several of them. This was a Pseudomonas, a very common gram-negative, common soil bacterium and plant bacterium that we put in the plant. And you can see, and it normally is a rod. You see it's spherical now. It's spherical. And the other thing to notice is these protoplasts are dividing inside these cells. They're growing. You can see a line of them there with this lower arrow. And you see the smallest one there, and you see the biggest one there. And I'll point out the smallest one is blue. That blue stain is a protein stain to show that there's protein in there. And we see the youngest ones have protein. The oldest ones are losing that. What happens is the superoxide goes in to through these membranes and will clip up the proteins in these cells and get protein actually out of those bacteria. So some bacteria are fully degraded. Some of them don't. Some of them aren't. Some of them survive through this process. But many of them will be degraded. And so they're constantly dividing to stay alive, to stay ahead of the degradation process. So this just is an illustration that shows a root cell. And you can see the bacteria that go in. When they go through the cell, when they go through the wall, they go in the cells at the meristem. What you have to remember is that those cell walls at the meristem are actively dividing and they're soft. So it's easy for these microbes to go through those cell walls. When the cells become mature, the cell walls harden. Okay, so, but at the meristem where these bacteria go in, where these microbes enter, they're soft, and so they can go in to those cells. And so they go through the cell wall, and then they get into space. Typically, it's in this space. We used to think it's only in this space outside the membrane, but that turns out that's not correct. They also go in the cytoplasm. They'll also go into vesicles, also in these cells, and in some cases, in many cases, in some cases, they'll enter into the nuclear envelope, to the membrane around the nucleus, which is 2 layers, and so they'll get in that. It's really interesting. But this shows what the bacteria look like in their rod form. This is with cell walls. And then with the reactive oxygen, it removes those cell walls, and then you get these little protoplasts, these spherical things, and these can be real tiny. They're not the same size as bacteria. Some of them are, but many of these can get really, really tiny, where you don't see them. It looks like tiny little bubbles inside plant cells. They're really cool, really, really interesting. There's a technical term for this, L forms. They've been known since the 40s or 50s. L form stands for Lister because they were discovered at the Lister Institute. So this is well known. It just wasn't known that they were used by plants as endophytic phases inside plant cells. Okay, so turns out the rhizophagy is really important because these microbes tend in this process really modulates plant development. Again, what you see, I've already mentioned that the microbes trigger root hair growth. Without the microbes, you have no root hair growth. The microbes also will trigger another behavioral characteristic of plants. We normally think plant roots grow down with gravitropism, right? They grow down with gravity. This is gravitropism. Well, if you remove all the microbes, they don't know how to do that anymore. They don't go down anymore. The roots will stay on the surface. Some plants, the roots will go in the air. The roots don't know where up and down is. It messes up root development without microbes. And those are examples. You also have root branching. You also have root growth, bigger roots with more microbes. If you have fewer microbes, you got fewer roots. And this works for biostimulant microbes. Biostimulant microbes do the same thing, and we've looked at several biostimulants. They do work. They do work. They're of course the best thing to have is really good, highly rich microbial rich soils. That's how to grow plants. And then you want good seed with microbes already in those seed too. Good healthy seed. You don't want any sterilized seed. This actually shows a Bermuda grass root where we remove the microbes by sterilizing the seed. And this was really easy to sterilize the seed and get these microbes off. All we do is soak the seed in Clorox, 50% Clorox, right? Just straight bleach, mixed half and half, put the seeds there, shake that for 1 whole hour, and that kills everything that's on the surface. It doesn't penetrate through the seed coat. So anything that's in the seed coat survives underneath the seed coat. But everything, turns out plants carry microbes on the surface and inside. So you can actually reduce them. So you see there's no root hairs there. There are little bumps there. Those bumps are the root hair initials that never elongated because there's no microbes there, or there's few microbes there. This is from the same experiment where we put microbes on it, and you can see now hairs. When we sterilized and then we put microbes on it, we put a Pseudomonas, a very common plant and soil microbe, and you see the hairs form right away. Yet immediately you have hairs. This is a bit older in the plant. You can see all these hairs, whereas the other plant was totally naked of hairs. And you can see the little brown dots in there. Those brown dots are the bacteria that are inside. All those bacteria. And this is actually a reactive oxygen stain. So wherever there's reactive oxygen around the microbe, then you're going to see this. You're going to see staining, and that's where the brown is there, right? All the brown. This shows these are the bacteria in the hair now, and you can see these protoplasts, these L forms, as they're technically called, but they're just protoplasts. They're just bacteria without cell walls. They have their membranes there, but and you can see they're staining there. They're loaded. Okay, and why is this? We think that the growth is because these microbes, this shows a root hair tip, and you can see the microbes there stained, they're accumulating in the tip. Those microbes in that tip secrete hormones, growth hormones. And the growth hormones that we can detect are ethylene and nitric oxide. Both of those are growth hormones. And when the nitric oxide is oxidized, you get nitrate. And because we have superoxide going on here, it's oxidizing these compounds. Both ethylene and nitric oxide get oxidized, but not before they trigger a growth spurt, an elongation. And when that root hair will elongate just a little bit, and some of these microbes will be ejected out. I'll show you that. This is that paper, by the way, that describes how this process is working. Okay, and this is going to show you. This is one of our experimental plants. This is a little sedge. I'm going to show you some slides now from this sedge, from this species of sedge. This sedge grows on these coral fossil deposits, limestone deposits, calcium carbonate deposits, little pits. Doesn't have much soil, but it has a lot of microbes in there. And this actually shows one of the root hairs of that plant. And you can see now, stained for the microbes, you can see these where this brown is. Those are the bacteria inside those root hairs. And I mean, that makes you think it's static, right? It doesn't do anything. But when you look, in fact, these root hairs are very active. And what you're seeing above is a living root hair. We didn't bend it, because if we bent it, we would break the cytoskeleton and we would stop the movement. But if we examine it, these root hairs, you'll see they actually are moving. And you can see here the shadows that are going around. Those are the bacteria that are in these root hairs. And so the plant is moving these microbes around in the hairs. It's processing these microbes. We think of root hairs as just absorptive structures, but in fact, they are microbe processing structures. And here, this shows it a little bit, and you can see the movement again. This is how they actually move. This is not much faster. It's a little bit faster than they actually move, but maybe about double, because when you look at these hairs, you see these microbes moving. And so you can see them moving these microbes to the tip, and they're accumulating in the tip, bouncing around, bumbling around there. And that's where they're secreting their hormone, growth hormone. And that's why these hairs grow with these microbes. This actually shows a microbes. You see the microbes that were ejected, but this fatness, this is mostly microbes in here, mostly those protoplasts that are inside the hair. Hairs are microbe processing structures. Root hairs are microbe processing structures. That's what they're for. They also absorb. I'm pretty sure they absorb, and there's work that shows they absorb. They are absorptive. They increase surface area, but before they are absorptive, they're microprocessing, or maybe during their absorption, they're microprocessing. So this shows actually a root hair, and it shows an ejection. Hairs there, and you can see the little pits there. You can see bacteria in the blue bacteria. They come out. You can see the little holes there. Those are the pits where those bacteria get ejected through that. And the plant is ejecting it because when that root hair inside grows, the microbes that are accumulated there in the tip get pushed out, get squeezed out of those pores in the tip, and so they go out there. I'll have an image that shows that. This is just a cartoon that shows that little diagram that shows it. You can see the bacteria. The bacteria here are red and blue. The ones in the tip I put as red. The ones in the side I put as blue. But you see, in the tip, they produced a hormone that causes this cell inside to grow. It grows. When it elongates, it forces those bacteria out those pits. These bacteria that are still in on the sides then are accumulated. They're moved around like that. That movement breaks these microbes up, increases their numbers, and then they accumulate in the tip again as that happens, as the continual movement happens. And then in mass again, they secrete those growth hormones that causes another growth spurt and another ejection. So when you think about this, plants take a few microbes in at the root tip, and they put out many, many more microbes from their root hairs. So they're essentially cultivating these soil microbes. Plants are getting them and increasing their numbers. So plants are really managing soil microbiology. This is the capability that plants have. This is the capability that we destroy when we don't have healthy microbial rich soils, or when we use fertilizers and too much fertilizer, too much chemical fertilizer, I should say, which truncates the capacity of plants actually to use microbiology. So that's how we take away the agency of plants by not taking care of soils and using too much chemical fertilizers. But this actually shows one of the hairs, and you can see this is a bacterium that comes from actually came from Japanese knotweed. So it's a dicot, right? A distant, and then we put it in this grass, in this Bermuda grass, a warm season grass, and it didn't quite work. Instead, these microbes went in. They're good endophytes, but they didn't eject properly. So you could see them in, and now you can see these hairs are banded, and that's where the growth spurts happen. The clear areas of the growth spurt, and at the tip is where the microbes, where it's dark, where the bands are where the microbes accumulated. So they didn't eject right, but you could see that it was this periodic ejection as growth spurts happened. You can see how that worked. This is tomato, and the root hairs of tomato, and you can see the actual ejection process of bacteria coming out. And if you look down at 3, you can see there's actually 3 little pores where those microbes are squirting out. I don't know if you could see it, but I could see that. There's little pores there. That's the point. And those are natural pores. They're not, the microbes didn't make those pores. They just happen because the root hair grows and you have those pores there. This is a nutritional function, and all of our evidence suggests that. Okay, so how are plants getting nutrients? Dissolved water, right? We've known that. That's why our chemical fertilizers work, because we put it in water solution and the plants can absorb it. Mycorrhizae, we know that plants, if they have mycorrhizae, they can get more nutrients. Okay, we know that. And now rhizophagy cycle. They can absorb bacteria, yeasts, algae into their structure, mostly bacteria, by the way, into their cells and extract nutrients oxidatively from those microbes in their cells. It's very difficult to sort out all sources, by the way. If we do experiments where we work with sterile soils, we sterilize most microbes from seeds, and then we put selectively microbes back onto, and compared to without microbes and with microbes, you see huge differences in nutrients. You see nitrogen, phosphorus, potassium, all the micronutrients are greatly increased versus plants without microbes. Okay, so we know the microbes are important, but of course that could be all kinds of things. That could be solubilizing in the soil, and also rhizophagy. Multiple ways that plants are getting nutrients from microbiology. So we've done experiments to look for specifically determine nutrients that are coming from rhizophagy. And I one of these experiments was where we labeled protein, bacterial protein, and then we incorporated that into agarose, and then we grew our some plants, in this case grasses, on that agarose, with and without bacteria. So, and this is what it looked like. So it's very much a laboratory experiment. Looks like nothing that you recognize from the field, but you do recognize the plants. And to the right is with minimal microbes, because we took the seeds, we treated them with Clorox to remove microbes, we germinated them, and you see they're growing in this bacterial protein agarose gelatin, right? They're growing there, and you see they're not doing much. There's not much color there. This is stained for reactive oxygen. So where it's colorful, there's more reactive oxygen. So there's not much going on. They're growing, but not much going on. Those shoots are a little smaller than here. Here, these seedlings had bacteria. These were not surface sterilized. And I should say that the bacteria protein that we used, we was labeled with nitrogen 15. And this is an isotopic nitrogen that we're able to track, that we can, we can send this material off to a lab and they can tell us how much of this isotopic nitrogen and this nitrogen 15 got into the plant. So, and this is basically this data here. And this, you can see when the microbes are there in the plant, you can see the plant's got this much of that nitrogen 15. Okay, when we remove the microbes, when we remove the bacteria, yeah, that's the microbes. When we remove the microbes, they got this much. So about 30% less. So according to this kind of experiment, rhizophagy cycle, or the microbes in this case, are worth about 30% of the nitrogen a plant gets. And this is not nitrogen fixation in the plant. At least we don't think this is nitrogen fixation in the plant. This is actually absorbing from the soil. So there's an increase, just rhizophagy cycle gives a plant increase in nitrogen. But we've done other experiments where we suppress superoxide and reactive oxygen in the plant to try to measure how much is coming through oxidative extraction from microbes. And what we do is we elevate carbon dioxide around the plant, and that suppresses superoxide formation. And it's superoxide that the plant uses to extract nutrients, to oxidize the bacteria, to get nutrients out. So when we suppress that, then the nutrients that fall in a big way are the divalent metals. Many of those are micronutrients, but also calcium, and manganese is one of the biggest ones. Manganese, there's like 700% more manganese when a plant can get make superoxide than when the plant can't get superoxide. But of course, separating all this out, all these sources of nutrients, is kind of challenging. And so this is really an open question now. We do know that microbial rich plants are also rich in nutrients like phenolics, antioxidant phenolics, and minerals, getting many minerals from the soil. They're more nutrient dense, as they say, and the minerals is one key piece of that. And we think that's rhizophagy cycle. So it's also possible to, and I'm not gonna go, I show a little bit of this, just a tiny bit, because it's relevant to things like compost teas and vermicompost washes, where you get microbes and you put them on plants. But we do microbiome transfer between plants, because you can also do this in one sense, making a tea from a plant, a ferment is similar, where you get microbes degrading plant material. A lot of those microbes are endophytes in those plants. So getting these microbes out of a plant, or getting microbes from worms, vermicompost, compost washes, whatever, put those microbes. In this case, we're using some of this corn that is microbial rich, and this is Walter Goldstein at Wisconsin that breeds some of the landrace corn, this microbial rich corn. So we can, for example, take that corn, grind up plant material, in this case leaf material, put it on seeds, sterilized seeds. And if you look over here, this is a grass seed without microbes where it was sterilized, and you see there's nothing there at the tip, and you see not few hairs. But here, this is where we put now from this corn. The microbes came from the corn, and you can see this area now. This is stained so that we can see where the bacteria are going in. This is the area where the bacteria go in. And you can also notice, you can see the staining there, bacteria enter there. The plant is cultivating those bacteria there. And you can see the hairs there. It stimulated hairs. Here, just another one. You can see the control. You can see now with the hairs there, you can see the dark areas where the bacteria are going in. The plant is cultivating those bacteria. They go in, and you can see the hair formation there when they go in. So plants are absorbing these microbes from the soil. This is where we used the POA. We transformed with one of the corn. The C46 is one of those corn plants, compared to non-transformed. And you can see there's growth differences between these plants. We don't know a whole lot about this right now. We're still tracking this process, but it may be a way that we can move microbes between wild plants and cultivated plants. But it also shows you what's happening when you use these compost teas and ferments and things that are microbial rich. Here's another one example. This is with Jerusalem artichoke. There's microbes cultivated in these leaf margins. These leaf margins are meristems, and in these meristems are where the bacteria are cultivated. And if you look at it, you can see shows the meristem of the leaf. You can see these dark cells. This is the meristem, actually, the dividing cells. You can see, and this is stained for nitrate. So we think there's nitrate there. We can't prove that at this point, that it definitely is nitrate in this produced here, but the stain suggests it's nitrate. And if you look closely at those cells, you can look like this, and you can see now in the nitrate staining, you can see darker and lighter areas. That's the meristem cell itself. You can see they're full of bacteria. The plants are cultivating. Here's a close-up of it. You can see the bacteria inside those cells, those black. This is nitrate staining again. The plants are cultivating these microbes inside their cells. It's hard to envision that there'd be another reason if it weren't nutrients. And in fact, when we do 15N analyses, we see in these growing these meristematic areas, that they're higher in absorbing nitrogen from the air. So it seems consistent that these have to do with nitrogen. We can move them in. This is into lettuce from that Jerusalem artichoke. This looks like it's actually disease protection. This is untreated. It's likely a disease that's causing these plants to spread out like this. Where we treated them with Jerusalem artichoke, what we did is grind the leaf up, and we sterilized the lettuce seeds with Clorox to remove the microbes. Then we washed all the Clorox out, and we germinated in the ground up juice from the Jerusalem artichoke leaves. And you see the plants do much better. They're not having the disease problem. I can't say that's nitrogen effect. That may be a defense effect, but defense is one of the benefits of endophytes. Okay, this just shows Jerusalem artichoke in POA. It didn't go in. This is that POA root, grass root, didn't go in. You don't see pigment there. You do see some bacteria in there. It looks like some little blue dots are there. Those should be bacteria, but not the Jerusalem artichoke one. Now here's chia seeds, right? And you can see the chia, which is another dicot. It went right in. Look at the purple there. All those bacteria that came off of that were around that area. So they absorbed them right there at the tip. So it's a lot of things that plants are getting from these microbes. They're getting development, right? Appropriate development. They use them to develop. Why? they've always had microbes. Plants are not free of microbes. From the very beginning of plants on Earth, they had microbes. We can look at mosses, the most primitive plants. They have microbes in them the same way. I mean, they don't have the same structures. They're not doing rhizophagy cycle, but they're absorbing bacteria from the soils. Okay, so what is that about? Our evidence says it's nutrients, that they absorb these microbes, rhizophagy cycle, especially they're getting nutrients from the microbes. Also, what happens is plants become oxidatively resistant because they have these microbes in them and they're using reactive oxygen to get nutrients out of the microbes and to control the microbes. Then plants upregulate their antioxidants, and some among the antioxidants are things like phenolics, which are also nutrients for people. And so plants, because they get these antioxidants, they become stress hardy, and so they're and stress any like heat is a stress. Uh, salt is a stress, so they become salt tolerant, they become heat tolerant, they become heavy metal tolerant. That's it. So endophytes, this is a long-term endophyte benefit that we know even fungal endophytes provide this kind of effect. Finally, with the microbes, the plants become resistant to diseases, and when those microbes are there, uh, fungal pathogens like fusarium or alternaria that normally affect the plant don't affect the plant as much or at all. In some cases, you'll have a little bit of disease, but the effect is minimal. You remove those microbes, and those pathogens eat the plant up. They'll destroy the plant. Okay, that's two things. One of them is the oxidative resistance, oxidative stress resistance that plants get, that makes them also resistant to these pathogens, because some of these pathogens are using reactive oxygen to try to initiate disease, make hypersensitivity reactions and stuff like that using their own reactive oxygen. But if plants are resistant to that, they don't respond. They're not bothered by it. The other thing is many of the microbes that become endophytic in plants in rhizophagy cycle, they actually will also colonize fungi. And when they colonize the fungi, they change the behavior of the fungus, and the fungus becomes less pathogenic. And this is, they're not inhibiting the fungi necessarily. Instead, they're going in the fungus and they're altering its growth rate. And they slow it down, slow its growth down. Maybe it's providing nutrients to the fungus. It's not 100% clear. That's our working hypothesis now, that provides nutrients. And that makes the fungus so it's less aggressive against the host. So there's multiple effects, but anyways, these are people in the lab doing much of this stuff, and I think I have a couple of minutes for questions. Do I? Anybody there?
Okay, sorry about that. We were muted, but we do have a couple of questions in the room here. So if you want to state the question, I can repeat it for Dr. White.
Yeah, I'm just asking about soil conditions like pH. How this impact the microbe and plant relationship?
So Dr. White, the question was about soil conditions such as pH, and how do those soil conditions impact the relationship between the microbes and the plant?
And we don't know the answer to that. And we don't know the answer to that at all. We've not done any of those experiments. Much of the work that we've done is focused on trying to understand the process, and we haven't gotten to that level of question yet. It's a really good question. There aren't too many labs working on this, so what we do what we can do, but mostly we're focused on some other questions. Like, well, I mean, like some of the things we talked about. But what you say about pH, pH and other factors, how does it affect? We don't know. There should be an impact, but we don't know what it is. Good question. Good question. It's unexplored science.
Another question that we have here online is that there's a lot of companies that are selling micro packs to add to the soil. and tests showing the shelf life of these microbes being very short, and that many of the microbes are antagonized by microbes in the soil, and therefore the results are minimal. Have you had any experience or done experiments with any of these micropacks that are being added to the soil?
We have not. We have not had any, but it is a real phenomenon, and we have noticed that some plants you can give them microbes, and they have their own microbiology, and that is quite intact, and they end up pushing out the microbes that we put in it. So there is that phenomenon. Microbe packs, we create these I'm talking for all of us humans, right? We create these um, uh, consortia, a mix of microbes, and we do things to them, different things to try to deliver them, and different mixes, and we create them not based on optimal use, but based on what seems rational to us. So oftentimes we'll put a, a, a nitrogen fixer, and we'll have a phosphorus solubilizer, and there may be some other a fungal disease controller, and put them all in there together, and then put those on the plants. So they're not necessarily really adapted to the plant, right? If we really want to get the ones that are adapted to the plant, then we don't do it the way we did it, which is just get the best microbes we can find instead from the soils or wherever they are. If we really want to do it right, we find the host plant, we get the microbes that the plant is using, specific ones, and then we put that back on the host plant. That's how to actually do that right. And put them on the plant, not on the soil. That's the thing. Putting microbes way out in the soil, is only valuable if they get to the plant. So really, this is a shortcoming of the way many of our biostimulant microbes are discovered. They're not discovered as endophytes. They're not already adapted to the plant. They're useful because plants can take them in the rhizophagy cycle and use them, but they don't keep them. They don't give long-term benefit necessarily. They're short-term. It's like a like a like a jump start, right? Kickstart rhizophagy cycle, and then you have to have a healthy soil to get them. So that would be yeah. So it's a real phenomenon. Yeah, they'll degrade in the soil for sure, but if they get into the plant, if they're plant adapted, then they'll stay.
All right, I think we have time for maybe one more question. Um, oh, we have one up here, Janelle. Go ahead, Alex.
It strikes me that maybe we need a 5R. We just need to add this to the mix. And my question is, part of that's getting the right bug in the place, the right crop. Uh, what is the longevity of this stuff? Is it something we have to manage annually? How much of it through the winters? Do we need to figure out how to sample for that 5R?
So the question, Dr. White, is, well, first it was a comment that maybe we need to add a 5R about microbial life. But the question is, what is the longevity of these microbes in the soil? Is it something that we need to be managing on an annual basis? Do they overwinter? What's kind of their life cycle?
I would say, yeah, that's a good question. Most of them are not gonna, the endospore formers will survive. Some of them can form endospores, but the plants are taking in. I mean, that's the importance of actually creating a living soil, right? That you want to have a soil with lots of life in it, cover crops and so forth, to keep the microbes alive. So if you do it right, you can have a good mixed community of microbes in the soil. If the soils are poor soils and you don't have cover crops, not a lot of organic material there, then many of the microbes are going to die as soon as you put them in. Uh, um, So, uh, but with the cover crops, it'll keep them there. And what the ones that will survive, by the way, if it's a bad soil, if it's a nutrient poor soil and microbial, uh, really it doesn't have much organic material there, not enough food for microbe. The organic material are the microbial foods, and also the cover crops. So if you don't have a good soil, then what happens is as soon as you put the microbes there, they die if they don't have endospores. And only a few of our biostimulant microbes actually produce endospores, and those are the bacillus. Many of the biostimulant microbes actually are bacillus because these will form endospores, and they'll lend themselves then to long shelf life and also survival in the soil and stuff like that. But we're missing out on some of the best microbes, and so that goes back to how critically important it is to also build up soil health. Good question.
Thank you.
Really good question.
So, thank you so much for your time and your presentation, Dr. White. And I'm sure if anyone has questions or follow-ups, they can reach you at your university email. So thank you again for your presentation. Really appreciate it. And at this point, we are going to move on to Dr. Josh McGrath, who is going to talk to us about the first tool. Dr. McGrath is with USDA ARS.
How's everybody doing?
Good.
All right, we have a list of research priorities for the USDA now, so we've got that set. Thank you, Melvin. And so actually, I'm going to start off talking about that before I get to my slides. I realize I have an hour of material, and I've only got about, 30 minutes until lunch. So I may just not do these slides. We might do something different just because I'm going to make Lindsay and Janelle really nervous and just say, let's throw first out the window and talk about something else. I can do it. I'll flip it. Sure. Yeah, I'm fine. So question I got when I came in. So this is the 4 R's. You got like one rainfall event in April, one rainfall event in May, and one rainfall event in August. Is that right, Melvin? Basically, it rained 3 times this year in Maryland, right? So apparently, since I left, everyone stopped using UAN and started just top dressing corn with urea. And the question was, how much of that urea is lost? What can I do to keep from losing it? Right? So I'm throwing granular urea on the surface in June-ish over top of corns. Air temperatures are in June this year pretty warm, 90-ish degrees, high humidity, ideal conditions for loss. 40 to 50% of that nitrogen is going to blow off as ammonia, right? Sitting on the soil surface. Is that about right? So what we recommend in that situation is NPPT. The kind of trade name for that most people recognize is like agricane, but there's a lot of versions of NPPT, right? Controls that volatilization of ammonia. But the trick with that is, so when I was asked this question at first, I said the solution is a center pivot, put water on it, right? That's what you can add is water. But if you don't have that option, NPPT opens up that window a little bit, but the problem is it doesn't help you that much, right? Because if it's really warm and humid, it's only going to give that control of volatilization for a day, 2 days, 3 days. Ideal conditions, cooler, maybe it'll give you control for 7 days and limit some volatilization. So you just don't have a lot of options if it doesn't rain or you don't have irrigation. That's just the reality of the situation. You're going to have loss, right? So thinking about the 4 R's and source, the way to go is to switch from that urea back to that 30% UAN, that 32%, that 28%. Interestingly enough, we use a lot of 30% where I'm from over in Delaware, what we're used to. And when I moved to Kentucky, no one had ever heard of it. The only thing out in the Midwest is 28 or 32, so that was a little weird. But if you switch over to that UAN, in that condition where the urea is losing 50% of its nitrogen, you might be losing 10-ish% of your nitrogen to volatilization. And so adding that NPPT is going to give you even a little bit more control. And that's where that source matters, combining that product, NPPT, with a different source, that liquid fertilizer. So that's your 4R talk. And I've got it working. See, I gave them enough time and they got it working. All right, so the next thing I need to do is acknowledge Dr. John Spargo up at Penn State. A lot of these slides are from John, I guess, some different talks as he's given on first over time. But let me see here. One other thing I wanted to do. I actually have to look this up. All right, everybody got their pens and pencils out? 240-962-2281. That's 240-962-2281. I'm not a psychopath, so I don't answer phone numbers that I don't recognize, okay? But shoot me a text message, and if you've got a question, you ever want to talk, I'm from the government, I'm here to help, right? So, all right, number 2 thing. If we're throwing the slides out, I've never done this online before, so we're gonna find out together if this works. Actually, let me see here. All right. Wow, that was good. Any of you ever listened to a Led Zeppelin album backwards? You know what that reference means. All right, we're going to do that. We're going to start with the last slide and go backwards. All right, go to soiltestfirst.org. Everyone plays with their phone during talks anyway, so if you've got a mobile device, go to soiltestfirst.org. All right, open it up. We're gonna walk through this together. This is literally the last slide. We're going to all do it together. The QR code is an interesting kind of Rorschach test. I noticed that when I was at the university, younger students would jump in a truck to back it up and hook it up to a trailer, and they'd look through the radio. People my age jump in a truck and look out the window at the mirror. It's the same kind of thing. I type in soiltestfirst.org, but everyone else is like kind of squirting the picture of the QR code. Everybody there? All right. All right. I've never done this on a mobile device. There's the 3 little bars in the top right corner. Touch that. Touch tool. Everybody there? Processing. We're going to test the towers. We're going to see what happens when we all try this at the same time. It's getting ready to get really data intensive in a second. All right. Click first decision aid. We're all there? All right, you see a map of the U.S. with a bunch of little dots on it? This is actually a slide in my talk. What I'm doing is I'm just skipping through all the slides. All right. Now, you're probably all already playing around, but let's try and do this together. Soil nutrient. Touch the little upside down arrow, and you get a choice of K and P, right? Let's do phosphorus. Everyone loves phosphorus. And for crop, let's do corn. You see all the crops there? In the drop down, everybody with me? There's a drop down, lots of crops. We're going to pick corn. Don't pick a state. We're just going to let it run all of the data from all of the states. But we're going to kind of select to what's important to what we got. Maryland in the room, I know. I see some Delawareans that I recognize. I'm a Delawarean, so that's strong showing. But we're going to let it go all states, right? And we're going to say, and we're going to do all soil series. Let's not worry about that. In 1955 to 2023, sure enough. Let's pick 0 to 8 and 0 to 6. 2 check boxes. That's the soil sample depth. Yes, sir. What's that? You don't see the map of the states. All right. Tax report is coming. I'm going to yell at all of you. Everybody can hear me if I walk around? I know there's people online. Sorry.
Oh.
3 bars. Now it's just, okay. All right, we're there. All right. So we've got 0 to 6 and 0 to 8. This is like never share the stage with children or pets or technology. It never works. And let's pick mailing free. That's what we use in Maryland, mailing free, but we won't worry about colorimetric or ICP. It doesn't really matter. And this is where the data intensive part comes. Right now, it's crushing Verizon servers. All right, while that winds up, we'll see what's faster, your phone or my phone. I'm sure yours is. Mine's a government phone. This is kind of loud. All right, I'm going to work backwards. Or actually, I'll jump back to the first slide. Let's jump around here. So that's what we're here to talk about, is that tool. The first tool. I'm at about 40%. Both my phone and me, about 40%. So, all right. So why first? I can fly through this pretty quickly. Don't need to waste any time. The idea with FIRST was to create this national database of soil test correlation and calibration to give farmers and their advisors transparency around where fertilizer recommendations come from and confidence in those recommendations, and to support future soil fertility research to really advance it and to get into where I work, precision ag. Everybody knows the cost of fertilizer is going up faster than the cost of crops. We're doing precision fertilizer. You know, the engineers did a great job of making a hammer so everything looks like a nail. We can vary fertilizer rate very effectively. I would argue that we don't have the ability to predict nutrient need at the same resolution that we can vary it with the equipment. I know Jared's giving a talk later about grid soiling samples, so I have some slides in here specifically just to troll him because I knew he was going to be speaking about that. You know, water quality, Chesapeake Bay, we all know this. And yet, most recommendation systems for phosphorus and potassium are based on decades-old soil test correlation and calibration. Sometimes, you know, I've been in industry, I've been at 2 universities, 4 universities, if you count when I was in school, and 5 universities. But like, we can count. And some people say, well, the land grant university's behind. The university's behind. No one else is doing correlation and calibration work. So if we're behind, everyone's behind. And I'm going to explain what that means in a minute. And there's this problem of coordination between states. So how do we make soil test recommendations? It's important to understand that, to understand what FIRST is here. It's 2 parts. First part is correlation, okay? So just to kind of explain this graph, you've got relative yield on the vertical axis, soil test on the horizontal axis, whether it's phosphorus or potassium, doesn't matter. Relative yield is unfertilized yield and fertilized yield. So I've got 2 plots, one with 0 pounds of phosphorus and one with 60 pounds of phosphorus. 0 pounds yields 100 bushels per acre, 60 pounds yields 200 bushels per acre. My relative yield is 0.5, I needed fertilizer. Generally, like 0.9, 0.95 is where most people kind of have that break point. So we're trying to find this break point of did not need fertilizer above the red line, needed fertilizer below the red line. And from that, we try and find this blue line, which is the critical soil test value. To the right of that, I don't need fertilizer. To the left of it, I do need fertilizer. So that's correlation. Calibration, these colors don't show up well here. So that's just to determine the sufficient fertilizer rate. That's a real key term, the sufficient fertilizer rate. That's just the rate needed to maximize yield. No one recommends the sufficiency rate, and we'll get into that. But it's the minimum rate required to maximize yield. Every single recommendation you've ever seen anywhere from anyone from any university was above that sufficiency rate, I promise. So that's where we get into plots. We take those 2 pieces of information, correlation and calibration, and we determine the recommended rate. And so this figure over here kind of breaks it down into build-up, maintenance, and sufficiency. So build-up is at low soil test peak, we recommend a lot of fertilizer to try and build soil tests up to that critical level. We're trying to get our soil test number to go up. I know many of you in Maryland and Delaware aren't trying to do that, and all you ever hear is about trying to get your soil test phosphorus down. But there are people in the world that need fertilizer, it turns out. All right. And then we've got this maintenance. And maintenance is crop removal. And it doesn't matter what soil test is. If you're yielding 200 bushel corn, the removal is always pretty much the same, right? Depending on varieties and little things like that. And then you've got that sufficiency rate. And sufficiency is always less than crop removal. And why is that? Any guesses? Why is sufficiency, by definition, pretty much less than crop removal? Alex?
Nothing operates perfectly efficient.
Nothing's perfectly efficient, and the soil is giving some up all the time. Even the worst soil, let's all talk about root microbes, root care. You can have a soil test of 1 part per million, 1 FID in Maryland speak. can still get phosphorus out of it by your plant. Plants are really good at squeezing phosphorus out of that dry sponge. Okay? So what a plant needs is sufficiency rate is the fertilizer. It gets the balance, the red that you see here from the soil. So sufficiency, if my soil test is, let's call it 20, it's getting I only need a little bit of fertilizer and I have enough soil to feed the rest. The purple part in this range, many universities are recommending extra to try and build that soil test. Is everybody with me? Does that make sense? Those three different types of recommendations? So this is philosophical. When you have a lab, any labs in the room? Someone from a lab? Good, I can talk bad about all the labs. All right. When you have a lab that says you need 300 pounds of P2O5 per acre, and the university says you need 200, the difference is in philosophy. They're looking at the same correlation and calibration data set and interpreting differently how fast they want to build to the critical level. And then when you get out here way past, see where my pointer is on the wall? And I don't need fertilizer to support maximum yield. There's plenty in the soil. There are universities and labs that still recommend some portion of crop removal because they never want to see soil tests come down. And that's a philosophical decision. And we can say we disagree or agree with that, but it's a philosophy, it's not science. This is one of my favorite little quips. I know you can't read this, but basically when I first showed up in Kentucky, 2015, so I left Maryland August of 2014. I'm cruising around on chat rooms, right? Soil science chat rooms. Soil scientist still. And I see this one, land grant fertilizer recommendation. This is like a national chat room. And basically, this person says, we've been using University of Kentucky soil test results for several years, faithfully, blindly, pulled a bunch of tests, sent them to a lab in Owensboro, Kentucky, sent them to UK, soil testing is free at the lab in University of Kentucky. Got 2 sets of fertilizer recommendations. They're very different. What do I make of this? And, I killed it. There we go. And this other person, I think his handle was actually Kentucky Farmer or something or another, was the handle. The only thing I like about soil testing is it's free, or it was. I'd rather pay A&L for better information. Waters is better. I found the same as far as Rex go. The first one was Kentucky Farmer. UK is really conservative to the point of hurting my yields. I also thought UK CEC numbers for my land were too high, usually in the mid-teens. A&L is around 10. That would be more in line with what I expect in this area. So this person clearly didn't like the University of Kentucky recommendation. A Kentucky farmer responded, I have no problem with minimal reps or maximum reps as long as the farmer knows what he is getting. And so that's what this talk is about. The farmer needs to know what they're getting. And if we can build that confidence in the recommendations that you as a trusted advisor make, then you're going to be more effective in getting those rates to be more in line with the 4 R's where they need to be, maximizing yield, minimizing environmental impact, maximizing economics, right? So those are our 3 objectives. You have a production objective. Definitely have to produce food to feed the world. We have an economic objective, which is entirely different and actually in direct competition with the production objective in most situations, and that's to maximize dollars per acre. And then we have an environmental objective to conserve soil and water and air resources, right? So when were current P&K correlations established or last validated? Part of the first was to go through and find this. And the group went through and found that about 75% or more are 22 years old or older. So that's how old the correlation and calibration studies are that support the soil test recommendations nationally. And this isn't a new problem. Back in 1965, there was a paper that came out, some of these problems saying that magic happens at state line. So I just grabbed a Google Earth shot of Maryland-Delaware line, or excuse me, Maryland-Pennsylvania line, a field that's split by the state line. If this field tested 10 on both sides of that state line, the recommendation per pound of P2O5 is different. Because magic happens when you drive across that state line. So back in, many years ago in 1994, the Stone Ages, I wasn't even born yet. Not true. Thank you, Emily. I have Emily on a laugh track. It's, there were papers that came out that said we need regional recommendations, we need current data, and we need it in a modern data system. And those were the objectives that first came up with. So the origin story, the idea was to, bring all of these things together. And when we first started hearing about this in the US, a guy from IPNI, which doesn't exist anymore, the International Plant Nutrition Institute, but they were the ones that invented 4Rs, right? They created the concept of 4Rs. And Paul Fixon, kind of one of my personal heroes, he gave this talk at the ASA meetings, the Leo Walsh lecture, and he said, basically, can we all come together and do like they did in Australia with the Better Fertilizer Decision Support System, where they bring all their data together into one dashboard that everyone can look at across state lines, to start synchronizing across state lines. And then Brad Jern and Phil Hess, Brad started giving all kinds of talks all over the country through 2014, advocating for this approach. And some friends of mine, John Spargo and Pete Kleiman at ARS, who was up at State College, Pennsylvania, they started kind of talking about this, and they kind of got me interested. So in 2015, we started having these discussions. Bromark used to host the oldest soil fertility conference in the United States. How many of you know, anybody know Howard Brown? Howard Brown, anybody remember Howard? He's out in Illinois, good guy. He always hosted it. They stopped hosting it, I guess, after he retired. I got to go to the last one. I moved to Kentucky. Howard called me up. He says, there's this conference. It's the oldest soil fertility conference in the United States. No one knows about it. It's like Fight Club. First rule of the Marcel Soil Fertility Conferences, no one can know about it, right? And they go up there and you fish for 3 days and talk about soil fertility. And Howard would mix and match who you were on the boat with. And I got to be with Giles Randall. And Giles and I sat there and talked about this project that I'm getting ready to explain to you for 4 hours while we fished up in northern Minnesota. So then I came back and brought some folks into Kentucky, and a bunch of other people kind of started refocusing on this. And now I'm going to take a little side story. Pete, that slide is for you. This is not another rambling with raft hunting story, I promise. We got this group together, and we got this phosphorus fellowship from IPNI. And our specific research objectives was to evaluate the spatial and temporal variability of the soil phosphorus critical concentrations in the corn production systems. We also talked about building a coalition to create a framework for a public database of P&K response trials. That was in the proposal as well. And so we wanted to look at precision ag and why we couldn't do precision phosphorus management. And John Spargo and I argue about this all the time. The way we do correlation and calibration trials is 5 rates of phosphorus randomized in 5 blocks at 4 locations for 3 years. We do this over and over again. Every 3 years, Nicole's going to walk into the Maryland Grain Producers Utilization Board, and they're going to ask her to recalibrate Maryland soil test phosphorus recommendations, or Paul or somebody to do that, right? We just keep doing it the same way over and over again because people say we need more data. But I would argue we don't need more data, we need different data. And so we took a little bit of a different approach in Kentucky, and we decided to build our own planner that could switch rates really fast going across the field. And we made these little plots that were 40 foot by 40 foot, 4-row corn planter. And we randomized in there 2 with phosphorus and 2 without phosphorus. And then we shotgunned those across the field so we can look at the spatial variability of response. And what we found was, on average, soil test worked. On average, we got a yield response across the whole field when we had a low heat field. But 50% of the time, at any one given spot in the field, it failed. So on average, when I had low soil test P in that field, my net increase in yield was there. But if I went to one plot, a coin was as good at predicting response as soil test. So if you want to do variable rate phosphorus and you have an average low peak field, you could just randomly pick where you turn on and off phosphorus as you drive across the field and do as good as soil test. Now, if you're going flat rate, soil test is really good. What you see is up here, it kind of tells you where I don't need phosphorus. That's the University of Kentucky line for where we say we don't need phosphorus. Obviously, I didn't have a lot of points up there. Everything to the left of that's a 50-50 split. The other thing that we saw is that as yield potential increased, the response decreased. So good dirt doesn't need as much fertilizer. some reason the plants were better able, microbes, longer roots, more moisture, whatever it is, the plants do better and need less fertilizer when you're in the good part of a field. Grid sampling. Jared, sorry. We'll still be friends afterwards, right? I'll buy you some barbecue at lunch. Grid sampling. For grid sampling to work, you have to have a 1/4 acre grid. And what I mean to work is to be able to make a map from that grid sample. If you're going 1 or 2.5 acre grids, that map is not reality, it's imagination. In other words, there's a spot I didn't sample, the map predicts what the soil test is, you stick a probe in the ground and actually measure it, you got a 50-50 shot of it being even close. And that's just math. It has to do with kind of the distribution of soil tests. But I still think grid sampling is useful. Don't make the map. Look at the distribution of soil samples and see where the good parts and the bad parts are, but it's the interpolation that fails. And the reason it fails, and the reason that universities have recommended variable rate for so long is because it seems to work. Every single field, this is a bunch of different fields that we did a bunch of 10th acre grid sampling on. We took thousands of samples at a 10th acre. And what we see is you have this strong, you have a bunch of high soil phosphorus, but on average the field is low. So you got a big bubble. So the average soil test for that field might be 20. But you got some samples way out here, right? And so once you grid sample, you're going to pick up 1 or 2 out here. And then that's where the rate drops, right? There's this random spot in the field with high soil phosphorus, and the rate recommendation goes down. But remember, all of our recommendations are above sufficiency. So just because you're fertilizing for this, and let's say the grid sample got it wrong, and instead of 20, you were actually 5, you're still above sufficiency, so you're not going to lose yield. So typically, when you grid sample, you drop fertilizer rate, we think that's a win, and yield doesn't change. So it appears to work. But the only thing is you're varying your rate in here, in the purple area, and you never get down here into the yield loss area, which is the green rate. So that's why grid sampling appears to work for variable rate. We need new approaches for precision ag, and that's what FIRST is about. I personally think it's total root length that's controlling this. When I got big roots, I need less fertilizer because the plant does a good job getting out. So we don't know for sure, but at the end of the day, we're going to need something besides soil testing for precision ag, whether it's mapping the soil, looking at landscape position, using climate models. Those are the sorts of things that scientists are working on today. Uh-oh. Messing this thing up. All right. So, see, that was not just another graph from the story. FIRST is going to support precision ag. How much time do I have left? I'm probably running out now, right? Okay. So the job of FIRST was to advance the accuracy of soil test fertilizer recommendations, soil test-based fertilizer recommendations. To create this national database, try and get some synergy across state lines, and to support best management practice regarding fertilizer. And so the first thing was to build a community, a coalition of the willing is what Paul Fixon called it, to pull people together to work on this. They went through, our chair right now is Nathan Slayton at University of Arkansas. We have regional, oh man, I keep hitting the wrong button, sorry about that, fat fingering this remote. We've got Matt Yost out here in Utah, Dan Kaiser up in Minnesota, John Spargo at Penn State, Dan Osmond out in North Carolina. And ARS is hosted on our servers. Lots of people involved. My former company, we funded a lot of the field trials. There's dozens of field trials around the U.S. And OCP is paying for all those field trials. And then there's a couple of other grants through NRCS and ARS. And then all of these universities and kind of NGOs and GOs are on here. And so there was a bunch of project activities and priorities to survey the recommendations and the methods. And so what we found out is that there's 8 methods for soil test phosphorus. Olson dominates in the west, Gray 1 in the north central, Malick 3 in the south and the northeast. So sometimes you hear about different soil tests, one being better than the other. It doesn't matter really that much, Malick 3, Malick 1 in Maryland. What matters is the data behind the recommendation, the correlation and calibration. Once you get into things like Olson's for a high pH soil, so you wouldn't want to use that in Maryland. We surveyed the states on what they claimed their recommendation was. 37 say sufficiency, 19% say build and maintain, 20% said hybrid, 20% say they use multiple recommendations. I kind of question these ones that say sufficiency. I've looked at all of them, and the one that's closest to sufficiency is Oklahoma, in my opinion. I think one of the big things that came out of that survey was reduced support for research. So this is the pitch that I'm going to make directly and unashamedly, is that as retailers, crop consultants, farmers, citizens of Maryland, Delaware, and Pennsylvania, you should advocate for more research. If you want better fertilizer recommendations, when you look at the decrease in the number of faculty conducting soil fertility trials in the United States, we've seen a precipitous drop since the 1950s and 60s. And so you need to advocate for those researchers in ARS, in land-grant universities, places like that. Looked at all the old soil test data back in the old days. Every university just kept it in filing cabinets and boxes like this. And so they brought all that together, put it into the database, and then started conducting new trials to add to the database. And the idea was to look at soil test correlation, so we're going to go back to the tool now, and they had to do some things like agree on a method for critical soil test value. So I showed this slide earlier, and you'll see that I have points above 1. First, I disagreed. I voted against this, but it's kind of a democracy. They decided to cap at 1. So they forced these points down to 1. I think that's the wrong way to do it. I think sometimes we do have lower yield in the fertilized plot than the unfertilized plot. A prime example, if you have too much phosphorus, you can have zinc deficiency and see yield loss. So I think that this creates some artifacts in the data. But they had to do things like agree on soil test correlation. They had to standardize that method. So they looked at all kinds of different methods and created this web-based platform. So let's go back to our phones for the last minute. Has anyone's phone gotten past 40%? We crashed first. Everyone give yourselves a round of applause. I'm done for the morning. The federal servers have just gone down. Is everyone stuck at exactly 40? 0. I'm stuck at 40. That's hysterical. Pete, you're the boss. Call somebody. You're in charge. Get on the phone with Bill Sachs. Yeah, my phone number's not going to do you any good. You got Bill Sachs and you're on speed dial. Call that guy. All right, well, I guess I'm going to have to actually use my slides. Oh, I was going to walk off stage, didn't you? No. So if I had crashed the servers by having everyone jump on at once. So if we go through and we pick everything that I just told you to pick, this is what you would have seen. See, I had a backup plan. It's not complete chaos up here. So you pick all that stuff, and you can actually drill down. There's going to be hundreds of trials. You can go right, here's the one I picked out from Wycomico County, Maryland, 1999. Who do you think did that trial? Frank Cole, maybe, what was my friend's name at the Lurie? Ronnie. It was Ronnie's trial probably down Wycomico County. There we go. It's like I have a terrible memory for names. So if you walk up to me and you know I don't remember the name, please tell me the name. But it's the side version of that story. But you can go in and look at the data. So he had a trial with 0, 13, and 54 pounds of P, but what he did was he banded subsurface before planting, and he broadcast incorporated before planting. Let's see, mailing 3 soil test was 44 parts per million. I'm surprised they had something that low at Wycomico, and there was no response to phosphorus. So that's going to be one of the points that you'll see when your phone stop loading. You're going to get a distribution of what all the yields are from 350 bushels to, that's some of my plots, 25 bushels. You're going to get all kinds of crazy fit statistics that I don't necessarily agree with. But what you're going to see, and this is amazing, my data is all about subfield data. Remember what that looked like, where it was 50-50 on the left side of the equation? Soil test is really good at telling me where to cut off. Up here, there's not much response. But down here, look how much scatter it is. Half the points are above the line, half are below the line. So when we look at whole field samples, and when we look at subfield samples, below the critical level, low soil test P, half respond, half don't respond. And sometimes you achieve max yield without fertilizer in these very low P soils. And that's because you've got good roots that can go out and get it. Maybe you've got good moisture, whatever it is. So there's some of these things that we need to figure out if we're going to do precision ag. And that was my last slide. So we made it through, and we have time for questions, or does everyone just want to eat barbecue?
I'm curious your answer on what I'm going to say later.
Besides funding, why has the number of university researchers conducting basic but necessary soil fertility research declined? So I think funding is the biggest, right? Just having the faculty to do it. And then I would say, and of course it takes a lot of resources to do that kind of work. And then if you want to do the more modern precision ag research, like I was doing in Kentucky, that's a tremendous amount. Wouldn't have been able to do it without fertilizer industry and corn grower support. That's who paid for that research in Kentucky. So the farmers and the fertilizer industry said this is important, right? So the money's the big thing. I think some of the other might be folks that don't want to touch it with a 10-foot pole when you get in this region, because it becomes political. If you're walking around talking about, hey, actually the soil test critical level, it's 50, Maryland says that you can apply fertilizer up to 100 FID. All the research out there indicates you will gain no yield above 20 FID. Folks don't want to hear that. So it's easier for a faculty member to keep their friends and not do the research, quite frankly.
Is there any correlation between SOM and yield response in a grid sample?
Any relationship between soil organic matter and yield response in a grid sample. There is a relationship between yield and soil organic matter when we look at it on a spatial. Now again, you can't interpolate those grid samples, but we were doing it fine enough that we could. And so in some of our nitrogen studies, we were able to see that better soil, where we needed less fertilizer, was higher organic matter areas of the field. So in Kentucky, we have topography, kind of like Western Maryland. And you get in those bottoms where you had accumulation of topsoils, you had nice deep soils, you were unlikely to need fertilizer down there. The side slopes with lower pH, thinner soils, that's where you needed fertilizer and you'd see that yield response. And the interesting thing was in that spot, right, exact same soil test of 5 in both spots, on that side slope, you get 160 bushels without fertilizer, 200 with fertilizer. At the bottom, you're getting 200 with or without that phosphorus fertilizer. You see similar trends with nitrogen, but of course with nitrogen, you're going to generally get a response. Although we did a lot of these trials with nitrogen, where every single plot, thousands of plots in a field, and every single plot had a 0 next to it. So we had the nitrogen rate and 0, all across the farmer's field. 25% of the time, I could maximize yield without nitrogen, and that was the really good dirt, right? But we can't predict where that happens. So I'm not advocating for not applying fertilizer. I'm saying we need to do research to find tools to predict where those areas of the field are, so as farmers we can take advantage of it, be more profitable. Yes?
You mentioned that having the root depth was a really important part of it. Is there any way with the sampling process that would maximize being able to figure that out on your fields?
When I left the university, that's what drove me out. I quit. So the question was, is there any way with the sampling that we can kind of figure out where that root depth is maximized and we can cut back fertilizer? We haven't found it yet. I'm hoping we can do some proximal mapping. I have high hopes with things like machine learning and AI. So we think it's related to landscape position, soil depth. There's some new tools coming out that might be able to map some of that, some of these new soil sensors that are proximal sensors you can run across the soil, similar to the old Veris in EC, which measures soil texture. And so can we combine all these data sets with these new computer approaches of AI to kind of look through the data sets and understand this area has a high probability of having big root exploration and needing less phosphorus or potassium. And most of what I said applies to phosphorus and potassium. One last note that I didn't make. Soil pH is different because it is log transformed. You don't have to know what that means. But remember when I showed that graph with the big bubble on the left and the tail that goes out to the right? You don't get that with pH. pH looks normal. It's a big bubble right in the middle and an equal tail on each side. So actually interpolation tends to kind of work with pH because it's not a skewed distribution.
All right. Thanks again, Josh. Really appreciate it. And so now, before we head to lunch, I do want to give the opportunity for our 2 sponsors that joined us with their booths today to just give us a brief sponsor spotlight. So I'm going to welcome up Tim Hushin with The Mill.
Thank you, Lindsay. And I'm Tim Hushin with The Mill. We are a family-owned, independent, full-service ag retailer. And my position, my umbrella within the company is managing the precision grounding side of the business, called Decision Ag Solutions. And we're trying to work with farmers that use their data to try and make more informed fertility decisions, also using some of the tools like Dr. McGrath had just talked about, kind of in season, making better fertility management decisions. And as Lindsay had said earlier in the morning, kind of when we kicked off, we've helped been a part of some of the funding and the grant work, very specifically the voucher program. And our growers have seen tremendous success from using those vouchers. So I challenge each and every one of you that are not wearing a farmer or an ag retailer hat to figure out how to continue those incentives directly for the retailer and for the growers, because they're a tremendous success and can actually help move the needle within our local communities. Thank you.
David Townsend with the Fertilizer Institute to come up and give us a few words. So for those that are not aware, the 4 R's of nutrient stewardship were actually first developed by the Fertilizer Institute. And so welcome, David, to give us readings.
Hello, everyone. And yeah, I definitely would love to give credit to IPNI and Paul Fixon. I was just listening to a podcast presented by Yara the other day about the origins and the history of 4R nutrient stewardship. And it's been really neat throughout my brief time at the Fertilizer Institute so far to see just the effectiveness of adoption and promotion and awareness of this framework. Like Lindsay mentioned, my name is David Townsend. I'm the Director of Member Programs at the Fertilizer Institute. And TFI is super proud to sponsor, in partnership with Mosaic, Corteva, ETS, and Yara, for our field days and symposiums like this one. So huge shout out to Mid-Atlantic for our Nutrient Stewardship Association and the alliances for your work in this area, and just happy to be here. So thank you.
Thank you. All right. So now we're going to move on to our farmer panel. So I'm going to ask our farmers to come on up and find a seat. And while we do that, we're going to do our next sponsor spotlight. So I'm going to ask Brett with Corteva Agriscience to come on up and deliver a few words on behalf of Corteva.
Thanks, Lindsay. I was sitting back there thinking about what I was going to say. Just want to appreciate the time we have here talking from the supplier standpoint. My name is Brett Burrow. I cover the Eastern Shore as well as New Jersey, and then run over here just south of DC and cover agriculture on this part of the world. From a Corteva standpoint, we provide nitrogen stabilizers, so they can then serve, and in the biological space, trying to provide innovative ways to be more efficient in terms of what we're doing with our fertilizer practices. So if you have any interest in that stuff, I'll be around here this afternoon to talk, and would love to hear about some ideas and trial opportunities going forward. Thank you.
All right, so moving on to what is generally my favorite part of these meetings is our farmer panel. So we do have some preset questions for the farmers, but we will open it up at the end for questions from the audience. As we all know, the Mid-Atlantic is actually a very forward region when it comes to U.S. farmers participating specifically in nutrient management and conservation practices. I actually got an email the other day from a colleague that's more in the environmental space. They were looking at the NUGIS system, which is the Nutrient Geographic Information System that tracks nutrient inputs and nutrient use efficiency. And they were like, why is the Mid-Atlantic region doing so much better than the rest of the country? And my flippant answer was because our farmers are better. But really, I think that we have had farmers that are very much leaders in both conservation and nutrient management. And we're going to hear from a couple of those today. So just to start us off here, I'm going to have each of the farmers just introduce themselves. Where are you from, and what do you farm? So we'll go ahead and start with you, John.
John C. Johnson from Stewartstown, PA. My brother and I operate what is the home farm for many years. I also farm other areas into Harford County. So I farm in both York County and Harford County. I'm still less than 1,000 acres, so small farmer these days. But we do grow corn, soybeans, wheat. We have about 60 acres of grain sorghum out this year. We do dabble a little bit with sunflowers, a little bit of hay, but that's mostly my brother. There's so much for handling the bales.
Trey Hill, I own and run Harvard Farms. We're based in Rock Falls. We farm in Kent, Cecil, and Talbot counties on the eastern shore. We grow corn, soybeans, wheat, barley. I guess our kind of what makes us somewhat unique is we're all regenerative, we're certified regenerative, so everything's no-till, everything's cover cropped, and then all the corn and beans are planted green into those cover crops before we terminate them.
I'm Steve Ernst from Appalachia, Washington County, Maryland, which is officially part of that. We're a long-term no-till operation that is a direct market, nationally distributed grain packer. And we farm roots and cover crops, and corn and beans, and barley, and wheat, and hay, and sunflowers, hogs and sheep, but our main crop is feed.
All right, Steve, I'm going to have you keep the mic there and ask each of you to just talk to us a little bit about when did you become aware of 4Rs, and when was it and what was it that made you kind of take that next step in your nutrient management journey?
So I come from a long-term no-till operation. We do cover crop planting, green, buy the same things that Trey is doing. And I also serve as a senior agronomist at Farms Elevator in Hagerstown, Maryland. And in 1988, Agrico Fertilizer Company, this came from the industry side, came out with Agritane, a product called Super Urea, which was actually an Agritane and DCD inhibitor combination manufactured proud urea product. And I was young and energetic, and I liked doing research a lot. So I set up a couple of series of 4x4 replicates of putting Super U in the row or off the side of the row versus urea, doing that, and then coming back and side dressing additional Super Urea over the top and additional urea. And so I was sourcing it and placing it, and the timing of coming back was much earlier than typical, about 8 inches, 10 inches, and looking at rates, and then looking at taking that to yield. And in those days, we did it with weight buggies and that sort of thing. And we saw significant yield response, and it was economic yield response. So that got my attention. And then as time went on, some of the no-till conferences in the Mid-Atlantic, which are world class, and some of the researchers that come in and talk about carbon utilization and that sort of thing. Some of the farmers from South America that would come in and talk about building an inch of topsoil a year with cover crops and things like that just really started to grab a hold of me on how can we take what we do as stewards of the land to a different level where we have an impact that is in the density of the food and in the enhancement of our environment.
Mine's a little probably simpler story. Mine probably came out of necessity. I got out of school in '98, and I think nutrient management plans started to get mandated in '99. I think was my first one. So just out of that, there was a financial necessity, thinking that at some point they're never going to allow us to put on more nitrogen and phosphorus per bushel. So the objective for me became, if I'm going to farm for the next 40, 50 years, how am I going to do that and stay financially viable? So we needed to really figure out where to place these nutrients, how to place them, and we started to do research on that. And since has, obviously developed from there, we're still trying to figure out nutrient cycling. If you had told me in 1999 that I wouldn't know any more than I know now, I'd be surprised. I really thought we would have had this figured out by now. It's the advent of yield monitors. We really started to get into precision ag. I had taken classes on it at that point, and I really don't feel like I don't feel like I've progressed quite to where I need to be. So I think there's still room for improvement. But a lot of mine came from that, just that nutrient management plan and seeing the writing on the wall that we needed to improve what we were doing. Not that what we were doing was bad, and I couldn't insult my father. He always did the best he could with nutrients and trying to balance them properly, but really starting to pay more attention to it so that we could then help facilitate that transition into utilizing them better. I'd say now we're concentrating a lot more on our nitrogen use efficiency, trying to figure out how to lower our carbon footprint, how to add more carbon into the soil, while at the same time trying to get prepared for like a scope 3 emission standards on the farm, hopefully within the next 5 years.
I guess I became familiar with the 4R program, according to what I heard earlier today in 2012, because the 4 principles certainly coincided with what we were doing on the farm. My first experience with cover crops was in 1949 when I was riding on the tractor with the cultivator. My dad operating a tractor, my grandfather sitting on a platform in the back, turning the seeder. They were seeding annual ryegrass behind the cultivator. Of course, the advent of synthetic fertilizers and so forth, people kind of got away from cover crops. My first experience with no-till was in 1953 when we finished drilling wheat. We had a little wheat in the old Van Brunts, so we pulled into the garden and just planted it for a cover. Of course, it came up just fine. So I could see there, wow, you don't need to work the ground. They're going to take my fun away because I always like to run the disc and the plow and all those kind of things. At that age, you wouldn't let me run the planter yet. They were pretty particular about the way the operator dealt with that. But as things progressed, planting green, I was planting green in the 80s because I had a cover crop already planted the previous fall. I sprayed with my planter at that time, and the cover crop got sprayed when the planter went through the field. So this planting green stuff didn't really seem like anything new. As far as conservation goes, my dad, grandfather were in particular about the way you treated the soil. You better not get out on that soil before it was fit. Just because you see somebody else out in the field, that doesn't mean we're going out on the field. So these 4 principles align themselves very well with what we believe to be the way to take care of our great natural resource.
Thank you. I think we're going to try passing this mic so that we stopped getting the reverb there. I apologize again for our IT issues. So the next question that we had is a little bit more specific. What is a 4R practice that you have implemented maybe more recently that's really worked for your farm, and why? And then if you have one, what's a practice that you tried that maybe didn't work for you? So what's worked, what hasn't worked?
Well, certainly 4Rs. They should have a song by that name, you know. Anyway, I don't know that any that didn't work. I mean, source, yeah, I like to vary the sources. Certainly, we use synthetic fertilizer. Also, we use chicken litter. Some farms we actually use biosolids, and then using synthetic fertilizer to balance out the potash need. Biosolids just don't provide enough potash. The timing, well, we do a lot of side dressing now. We didn't used to, so I would say that really got us into the mood there. But hey, I really had it in mind before, but hey, if this organization was promoting it, hey, it must have some merit, so I will go for it. The placement, every planter and drill that we have has in-furrow capability. And we like to put not a lot down in furrow, obviously, but some nutrients in furrow to kind of enhance the germination and the seedling growth.
I'd say our biggest success has been variable rate nitrogen. While it's not completely new to us, it's something that we're always fine tuning and getting better with. And then kind of the success slash what's working, not working is trying to use that variable rate nitrogen in conjunction with the biologicals that are providing nitrogen enhancement, trying to lower our use of synthetic nitrogen, but also figure out, is there really an environmental impact if we do that through the substitution of biologicals? So the success, I would say, is the variable rate nitrogen in side dress, trying to get more of that nitrogen pushed towards the side dress. One of our failures was when we didn't put enough nitrogen up front. There for a while, I got to where I was like, oh, I can really start to push a higher and higher and higher percentage to side dress, thinking that was the right move. And then we were starting to get some decline in our seedlings. So we've kind of backed that back towards the starter fertilizer, up in those nitrogen rates a little bit, and then still doing the variable rate side dress. But I think that's probably where I have the most room for growth. One of the other things that we're struggling with is trying to vary the rate of chicken litter and compost. We have the equipment to do it, but it just seems like we're really struggling with the technology in order to get it implemented. We're basing it on grid samples, which I don't thoroughly agree with that, but I don't really know what the alternative is in order to really ascertain exactly how much phosphorus is in the soil and where it's worth investment in the technology to get it to where we can really start to vary the rate of the chicken litter in the compost, which I think is something that we really need to be doing moving forward.
So probably back in the 90s when we went, and let me preface this, where we farm defensively. So I farm one big rock, and it sticks up a bunch of places. And we'll spend $20,000, $30,000 knocking rocks off the field. Now we crush that and use that for gravel, but the point is we farm from a clear different perspective. So everything we're doing is defensive. So the placement of fertility has been perhaps the biggest advantage to us in the last 20 years. I will do plot work before I decide that I'm going to do something for sure. That's just the way I think, even though I'm not going to tell you I'm that analytical. So when we did our replicated plots for pop-ups, in furrow pop-ups back in the 90s, we saw a big difference, and we keep a small 8-row planter set up to split applications. We can run 4 rows of a treatment, 4 rows of another treatment, so we can check and look at different materials in that trench to see if we get a response to it. And there are lots of different things that we're looking at, but I forget who said this morning about we're growing roots. And we've been looking at that really hard the last number of years. I tend to go lower tech as long as I can. My boys are much more oriented toward the better technology. And they look at things from a different perspective, so we have different things going on. But just looking at roots and how roots and cover crops work, how they move nutrients and recycle them, and then looking at where I place the fertility with the planters, and back in over top with the trucks and the high boys, is kind of a lot of the things that we're looking at, and placement in the environment where you're purchasing inputs, whether it's litter or synthetic fertilizer or whatever you're purchasing, or you're utilizing them off farm from the things that you produce. It's really important to know. And a lot of times, Josh was saying about we have all this data, but we don't have the right data. He's 100% right. There's gobs of data out here, but it doesn't necessarily apply to the microenvironment that I farm in.
So Trey, I'm going to go back to Trey for just a second here. So Trey, you briefly touched on this But what have been some of the barriers to implementing a new 4R practice? So you spoke about the equipment for variable rate poultry litter application as an example. What are some other barriers that you have to trying new practices?
A lot of the practices I find that come out, the key to farming is very fundamental, right? It's getting the seeds in at the right time, the right seeds and the right things. But efficiency, planting date, and getting on the fields and being able to work as fast as we can with the time that we're allocated is usually the most important thing. So to me, that always trumps pretty much anything. So folks will come to the farm with a new technology or something, and then all of a sudden the planter's sitting for 2 hours, and I'm like, this is unacceptable. We're supposed to be able to plant our corn in 10 days, regardless of the size of the operation. But you're supposed to be that efficient according to the university. Some are actually recommending 8 now. So 2 hours all of a sudden is a big deal. So the biggest problem, one of the biggest problems we've had is getting the technology, not just getting it on the planters and getting it in the equipment, but then once it's on, I don't mind putting the time in at the off season during the maintenance time, and we'll get prepped and we'll get a plan together. We'll be ready to execute that plan. But a lot of times the plans fall apart once you get to the field. Getting communicated with the sprayers, getting communication tied in, so it's on, so it's in the air, all of that stuff. So it's in real time. It's often very difficult. A lot of times we might be side dressing on a weekend. So if I'm depending on a third party that doesn't work weekends, I'm not going to stop the sprayer because I'm waiting for somebody to go to work. So all that stuff we've had to keep in house, which then presents its own set of challenges because you're trying to employ personnel that have that capacity, but can also do the normal work of day-to-day farming. So that those combinations of people are often hard to find. So I'd say that's probably been our thing. Or you'll depend on one supplier, and then the price point will change with that supplier. So you're using that supplier for a specific technology and a specific service. But then if you have to purchase it somewhere else, a lot of times that tech and support doesn't transfer with that purchase as you switch suppliers.
Yeah. What about you, John? So you have maybe a little bit of a different perspective as a smaller farmer. What have been some of your barriers to implementation?
Hey, I don't know really. I don't feel I have many barriers. I'm willing to try a lot of new things, and some work, some don't. But hey, we need more encouragement from our government. Hey, the state of Maryland does a great job promoting cover crops. I mean, we all know that sitting here. And some of the other mid-Atlantic states should follow suit, which certainly would help with the bay cleanup that we all hear about. But hey, nobody said I couldn't do furrow. Nobody said I couldn't side dress. Nobody really told me when I could apply. And hey, I could use synthetic fertilizer. I can use chicken litter. I can use biosolids. I don't feel limited.
Awesome. So Steve, I'm going to come back to you to start off with this question, and you touched on it briefly in your introduction. But how do you evaluate whether or not there is an economic return on investment on implementing 4R practices, and what kind of returns or not have you seen?
Well, I have my general manager of farming operations is my eldest son, and he's an ag economist. So you know what that means. And it's funny how, we're a generational operation. There's only 3 generations on the farm right now, me through my grandchildren. But we've been there a long time. My grandchildren are the 8th generation. And it's funny how you spend your lifetime doing things a certain way, and then you figure out, well, maybe it wasn't the best way, and you got to be willing to change. And it's really good to have people who can crunch the numbers for you. Surrounding yourself with those kind of people. We have a CFO in the office. We have an office manager, and between them and my eldest son, they're going to look at the numbers really hard, and they're going to tell me, my son will say, Dad, that doesn't pay enough. I don't care if you like doing it. Sorry about that. And it's like, sometimes you just go with that, because the ultimate goal is to make sure that my grandchildren can have a viable economic unit. That's kind of where we're at. We tried a lot of things, and we do a lot of things that don't make sense just because it works for us. We do a lot of our own building of planters and grain complexes and all that stuff in-house. just because a lot of that stuff is gold plated out in the industry and I can do it for a lot less. And also in my conditions where I deal with environmental situation of limestone rock that is very much going to break your equipment, not where I just break it. We have to look at things differently from a different perspective. So we're going to do plot work and run numbers and that sort of things. So one of the things that hasn't worked for us has been the foliar feed thing. We've looked at that a number of times here in the last 10 years probably, running a bunch of plot work, crunching the numbers, trying to see, does it pay or doesn't it pay? We are pretty sure that adding certain things to the tank mix when you're going over your crop make a difference on stress to the crop, but they may not necessarily be fertility. And some of that stuff is really wiggly until you start going to replicates and trying to get a handle on it. But just remember, those replicates are at that specific place at that specific time. It's not, we can generate data and say, well, these were the results, and next time around it may not be the same results. So we have to be very careful when we start saying, well, this is the measuring stick that we're going to measure from. And one of my pet peeves there is soil organic matter. We build soil organic matter and we see it go away, and we don't change our practices, and the soil health people tell me to implement cover crops and no-till. We've been doing that a long, long time. So we have to be really careful as industry and regulators and things like that when we start saying this is the answer, because it's not always the answer in every case.
Yeah, Trey, so maybe to dig into your specific operation a little bit more, how do you evaluate new practices, right? So we're already, like Steve said, we're already doing all of these different things, cover crops, no-till, nutrient management, baseline. How do you look at the additive return on investment for additional practices?
But $7 corn or $4 corn? $7 corn, I try everything and anything, and then we overlap plots and we do all kinds of stuff. This year it's going to be NP and K.
That's a good point, though.
Yeah, I mean we're gonna knock out a lot of that stuff because I don't think that the margins are so tight on a lot of it. A lot of things will run if we think it's better at a break-even point. I don't mind running a biological if I know it's going to work 50% of the time, and I'm happy to do it. But this year coming up, we're probably going to be a lot more risk averse to any type of put forth any budget towards that kind of thing. I'd say we're similar to Steve, but not quite as analytical, a little more fly by the seat of our pants. I try to do big plots. So if it's something, as long as I know there's not going to be a damage to the crop, we'll do it on a lot of acres, and then we'll just do it one-off with a sprayer, try to isolate that product. Then we'll come back through and check the yield monitors and see if we got a return or not. Fungicides were probably our first big experiment with that 25 years ago. In some years they work, some years they don't, but in time, we realized that, it's one of the things that I struggle with as a soil health guru is we still apply a lot of fungicides, but I always get an ROI on it, right? It's a really easy one. It's just a matter of which one you choose. The biological space, I'm having a lot more trouble getting those consistent results that we did out of the fungicides. And I'm having trouble seeing it in the fields. A lot of times you can see eyewitness, just drive-bys, you can see a difference. And if you can see a difference, it's doing something. So that's kind of the way we're running. Our biggest struggle lately has been as we got more into planting into bigger and bigger cover crops is figuring out the nutrient cycling and then trying to figure out what credits we can give to the legumes. All that stuff to me is still a little bit of a mystery. And it's really hard to put sound science to it because we don't really know what's out there. And I think once we can start to really photograph the fields and get an idea of what's out there, then they come up with AI or algorithms to help determine how much nitrogen was there from those legumes or what the carbon to nitrogen ratio was of that cover crop mix. Then we can really start to develop a much better, more precise way of putting our synthetic nitrogen down. I don't know if that answered the question.
Yeah, no, absolutely. And John, did you participate in our split nitrogen cost share program?
No, but I did have a voucher for running the dual over a couple farms where the electric conductivity was recorded, and we were able to make zones out of those fields it doesn't go by field border. It goes by the type soil and particles and so forth you have there. So we have done that. We have variable rate applied nutrients based on what the Dulam work did for us. What we do for side dressing is we will superimpose then the yield maps over the Dulam maps and then apply, as we hope will give us the best economic return. I do have a master's degree in ag economics, so I should be doing a lot of analyzing. First thing I do is look at the balance in the checkbook. And from that, I said, well, this new idea, maybe we just better put it back a year. As prices have already been mentioned, there's not too much room to play games right now. But when prices are good, we're much more willing to try new things or adapt to old things that we hope will either increase yield, or that's not the only thing we look for. My brother kind of likes to keep the expenses down as much as possible. So hey, if we can eliminate this particular application, can we eliminate the herbicide? Because we have cover crops planted, and the biomass from the cover crops has suppressed a lot of weeds for us. In fact, the one in particular I think of is the rye on mare's tail. I am really impressed with how much suppression rye can give to the control of mare's tail. So keep the expenses down and the yields up.
Yeah, I'm going to stay with you for just I'm going to stay with you for just a second here, John. I want to make sure that we get to this question and maybe one more before opening up to the audience. So you've all kind of touched on this in your own different way, but can you tell us a little bit about what your overall soil health goals are and whether or not you associate those soil health goals with your 4R program?
Soil health, what that really is soil life, right? So what we try to do is promote as much microbiology as we can in the soil. Of course, we think that cover crops are a big part of the answer. But now, looks like the new wave is the biologicals. And we have played around with the biologicals some. I can't give you any figures as to whether they've really made us any money, lost us money, or whatever. But I truly feel that this is something we have to pay attention to. Of course, we have the biologicals, and then we have the biological stimulants. So, where do we go with that? The biologicals themselves, you do have to pay some attention as to how you take care of them, how you store them. The biological stimulants, not so much. They can sit on the shelf for a while, pull them off the shelf, they can possibly do the job they're supposed to. So, my goal is to continue to improve that soil life. Why? Well, better the soil life, the less synthetic fertilizer we're going to need. Tim likes to hear that, yeah. The root extension down to that biological life is what we'll promote, what we feel are yields for us.
I guess my goal for the next 5 years would just be establish a baseline and figure out what it is. We've been working towards it, and we feel our soils are much healthier. We do earthworm counts, different things, but I still really feel like I'm flying blind. I don't really understand what soil health is. We look at organic matter increases. I had an intern do a study of all my nutrient management plans on 3 fields over 24 years. And he graphed and did, really careful analytics on organic matter increases over that time, and it showed a positive increase in organic matter. So that to me was saying that we have healthy soils or that our soils are building. I think that synthetics are probably poor for soil health. Organic materials are probably better. So how do we start to limit them? but I still don't understand the impact of either, and I don't really know what a truly healthy soil looks like. So trying to figure those things out. We've done a lot of experimentation with the 3 different companies that do the DNA soil sampling that claim to tell you what soil health is. So they've got Pattern, Biomakers, and Trace. And all of them come out, but I'm like, they're like $125 or $150 sample. So it's like, what's the frequency? What's the time of year? You know, if we go back and sample year on year, are we going to detect any differences, or are we just going to detect what's there? One common theme was that we had a lot of disease in our soils. So I'm like, well, is that a sign of a healthy soil, or is that a sign of a non-healthy soil? You know, I like to think that we're going to achieve this magical balance where I don't need anything, but then I'm like, well, Mother Nature really wants things to break down quickly, which would be disease-ridden. So I don't know. So that's my goal. We're trying to make soils healthier. We're no-till, we're cover cropped, and we can see a visual difference. We've got aggregate stability, we've got mycorrhizal fungi, we've got all these things. But in terms of a measurable and deliverable that I can take to someone to demand a premium, I still haven't figured that out.
Yeah, I can concur with Trey on trying to figure out what the baseline is or what the metrics are. It's really difficult, and it's very frustrating, I think, because, you know, every place is kind of different. We dug some pits. We had a cover crop field day last fall and the spring. And this spring we dug some pits in a field that was probably one of the last fields that I can remember doing tillage in back in early 90s maybe, when the zone builder came out, and that was a big fad thing there. It didn't work in our area because it broke. But anyhow, so I was basically, I tell most people, my friends in the Midwest and things, I farmed about 3 inches of topsoil. And I was really pleased to see where we dug these pits. Underneath that topsoil, there's either limestone or orange clay and then limestone. In that particular field, we dug those pits, and we had about 8 inches of topsoil there. So that had been in no-till and cover crops since the early 90s, and we probably picked up 5 inches of topsoil is what I would say. So when I hear people talk about, you know, really increasing topsoil and soil organic matter really quickly, I don't disbelieve them, but I do have a male bovine defecation indicator in the back of my head. And sometimes it goes off, and I think that, you know, figuring out what's that legume doing for us, how much can we cut back? The plot work I did when I went to certain legumes, and all the legumes are different, indicated that I didn't need as much side dress because I was getting a later breakdown of nitrogen through the season from that biomass. So there's all kinds of things, and obviously I think I'm always focused a lot on soil organic matter, but even sampling, doing your soil samples in a really dry fall are going to affect soil organic. And sending off to some of the soil health labs, and they're very proud of those tests.
Good tests.
Yeah, they are, but they're very proud of them. And if you got a bunch of samples to do, it gets expensive, and it's kind of, you know, that's a little bit of a dead expense if all it tells you is to keep doing what you've been doing. So again, I think trying to come up with making sure that we don't try to establish metrics from the regulatory side that are going to punish, penalize, and set people back. Because the benefit to the cover crop and the long-term no-till and the planting green is that you're armoring your soil. So when you get into these stressful years like what we have this year in our neck of the woods, we had severe drought. You're going to have a better looking crop out there, and it's going to give you a better crop in the end. But it's not just that. I mean, we've gone to 20-inch corn. We've done a lot of different things, not just the cover crop and that. We run stripper heads in the small grains, so we have more soil armor, more lower soil temperatures because we're armoring the soil, that sort of thing. And do those things help alleviate burning soil organic matter? Obviously, no-till does. But how do we measure all the rest of that? And there's all kinds of science, and it's really good stuff. And there's all kinds of data, and I can't sort through all of it. But everything isn't as exact as we'd like to think it is, because I can't control the weather.
So I want to make sure that we have time for a couple of questions from the audience. I have some additional ones here, but wanted to make sure that you guys had a chance to ask any specific questions that you might have. Questions in the room or online? The question is, are any of you participating in carbon markets?
I sold carbon credits in 19, and again last year. The company I was dealing with, Nori, is no longer in existence. So it went well. I think it's a stepping stone towards insetting. So I'm working with two companies now on a regenerative branded product on barley and getting slight premiums for that. And they're looking to model the whole farm, but it's not actually it's not a consumer driven demand as much as it is through the commodity federal program is where the premiums are coming from. So I think it's coming. It's just been, I was anticipating having this 5 years ago instead of just kind of getting started into it now. The carbon markets seem good, but I think they're getting taken over by the big companies, and I don't know that there's a whole lot of money for the farmer in it. $8 or $9 an acre, I don't think really changes anyone's practices.
I have not participated, have not sold any carbon credits. I am currently in touch with Mitchell Hora from Iowa, and he is big in carbon intensity. And the only thing is that has a lot to do with ethanol plants nearby, and I don't know where the closest ethanol plant is here. Now, some of these credits can be traded and bought, sold, and so forth, but this carbon intensity deal is definitely in its infancy. I'm going to pay attention, but I don't know if it'll play out the same way the carbon market did for me or not.
Guess I'll have to be careful how I say this. I'm not sure you can pay me enough so you can pollute. And I'll just put it that bluntly. So back when this whole thing started, I don't know how many years ago when we started talking about carbon credits, we had I think it was NRCS was doing those analyses on your farm, that kind of thing. And we've kind of taken the approach that Trey did. He's looking at a branded regenerative product. We are a branded regenerative product. We are Wholesome Harvest, Ernst Grain Livestock. We market every acre that we produce in 40 and 50 pound bags, and we have a number of growers that grow for us. So we have taken that approach, and that basically my mindset has always been, because I'm an old hog farmer, bring me something, I'll figure it out and do it myself. So I have positioned our business to capture value by doing the branding and selling a superior product. We are a non-GMO producer, but I am not a philosophical non-GMO producer. I'm a non-GMO capitalist. And what we've done is we've developed a non-GMO product, and that gets us in the door with our clientele, and the quality of our artisan feed manufacturing shop keeps us there.
You all are on the cutting edge of innovation, conservation, productivity, sometimes on the bleeding edge. How do we reach the other folks that aren't? How do we help the people that aren't as far along as you are? You ask them the same questions and looking at their operations.
So the question is, obviously we have some of the more cutting edge farmers here, although I would argue that there's probably more in this category than some people would think. How do we help those folks that are not quite to your level continue on the 4R continuum?
Well, number one, you need to be a lifelong learner. So that means that I have to be willing to continue to learn. And as you learn that you know less. But anyhow, encouraging and getting your contemporaries to get to field days and events where these kinds of things are talked about and shared, And there are a lot of organizations in the Mid-Atlantic because the Mid-Atlantic is, in American agriculture, the Mid-Atlantic is the cutting edge of this. You know, yes, there's some guys in the Midwest doing some tremendous stuff. But, you know, there's people out here that have been doing it a long time. John was talking about how long, and, you know, no-till originated in Kentucky, right, Josh?
I disagree. I think it started in Maryland.
Yeah, I tend to agree. Maryland and Pennsylvania, York and Franklin and Adams County and Maryland. I mean, and it was for different reasons. It was mainly soil conservation. For us, it was rocks. You know, we just didn't want to pick them up all the time. But the whole thing is getting your contemporaries to buy into the fact that there are things that they can do that save them money and have the opportunities to capture more profit because of not only saving them money, but marketing and such things. I think that's the thing. You know, the biggest part of the word agriculture is culture. It's a cultural change. People in the Midwest, they ask me, well, how did you learn to do this? And I'm no genius. I don't know any different. That's the whole thing. I just don't know any different. You know, so we've done this a long time, but it's cultural. When I was in Iowa the other spring, 2 or 3 springs back in corn planting time, I was astonished that I saw one field of grain. And that was eastern Iowa, where it rolls like York County, you know, and Carroll County. So the biggest part of the word agriculture is culture. We've got to change the culture. The only way we do that is if they see success from the people who are on the cutting edge, which is the bleeding edge sometimes.
Yeah, I'd have the same answer, just maybe articulated a little bit differently. I think that we need multiple funding sources in order to enhance the adoption and practices. The funding source, if you look at, you know, everyone says, oh, farmers don't adapt, and they don't adopt, they're these old, you know, curmudgeons or whatever. You look at Roundup Ready technology, it was adopted in 2 years, fully. You look at cover crops in Maryland, what are we, 80%? I don't want to say the number, whatever it is. Why do we have such a high adoption rate for cover crops in Maryland? Because we're paid to do it. So if you could get into carbon markets, I didn't fully believe in carbon markets. I still am a little bit of a speculator, but I did it because I knew that was an extra funding source for me that may lead to other people getting that similar funding source. And of course, the first person that gets paid the most typically. But I think if we got those different avenues where you have cover crop money, you have insetting, maybe you have a consumer that needs something with a lower carbon footprint, all of a sudden the farmer starts to get the cover crop money plus another incentive. Maybe it's an ecological enhancement money, maybe it's environmental money, maybe it's for the person that's consuming the corn that goes into the chicken, whatever it is. If we could get into that $100, $150 an acre range, or even similar to what Secretary Addix was talking about, having it where we would have a place we could go to actually see all the different programs in one place. I get confused by them. I find it hard to get in and get them done. And then also on the other side, a more micro concept is that I don't really know of anyone that's really doing really good economic research. You've got 2 economists and the father of an economist here. Am I making more money than my counterpart that works all the ground and doesn't do the cover crops? I don't know. And I'm hesitant to recommend it to other farmers because I might not be making as much money as other farmers. I'm probably not. I mean, I'm sure there's some that are, but am I the mean? Am I below the mean? Am I 90th percentile? I have no idea. And I don't really see much good research coming out on that where they have actual farmer expenses and not just, you know, oh, we call this is the retail price of the Corteva products when no one's paying retail. So I think until you could really drill that down, figure out what is the yield implication of doing no-till and cover crops versus conventional tillage. In a good year, I take a yield drag. I mean, I'm convinced of that. Soybeans, I think I get a bump. But when the last 3 years of corn, I'm not growing as good a corn as my neighbors, and it upsets me. And I mean, it does. I mean, that's an economic loss to me. And if my competitors are doing better than me, that's not good. So it's, you know, I'm under the understanding of like, well, I'm going to try and fix this, but they're seeing that. So I'm sure they're like, well, why am I going to farm like Trey when I'm growing more corn than him? And they are probably right. So research on all of that would be imperative. I think a lot of the universities need to do more within the regen space.
I want to make sure that we also get the opportunity for each one of you to kind of leave this audience with your closing message. So we've talked about a lot of things, but in the room we've got mostly agribusiness, technical assistance providers, government agencies, conservation professionals, universities. So if you as a farmer could leave them with one message around, farming in 4 R's, what would that be?
Well, first of all, let me say, I'm a very poor salesman. I probably would have gone hungry if I depend on my ability to sell. Cover crops, 100%, we're 100% cover crop. We have been for many years. So as far as selling this to my neighbors, they see what we're doing. They like it, go ahead. They don't like it, I don't try to push it on them. I've spoken in St. Louis, I've been to Hollywood, Florida to promote cover crops, but hey, I told them what it did for me, but I'm not trying to pretend it may do the same for you. As far as my message, maybe this is getting a little off, but diversity is what I preach. I'm talking about diversity of the cash crops. I'm talking about diversity of the cover crops. I just finished planting behind wheat a cover crop mix of 8+2, 8 species out of the main box and 2 out of the legume box for a total of 10. Why? Hey, I want to provide those microbes in the soil with various root exudates from various species. And I think that would generate as much soil life as I know how to do.
Coming from a depressed farmer due to a drought, prices, I'll try to be optimistic. I think in agriculture right now, it's never been more exciting. With the technology that we've got coming out, the reliability of the machinery. When I first got out of school, I was more or less a mechanic, right? I mean, that's all I did was work on equipment, drive equipment, work on equipment more. And I think now that the equipment's become more reliable, we're getting into the technological space, we're getting into AI and all these things. I think that we've got satellite imagery, we've got sprayers that can do see and spray, we've got all these things. And now with all these new tools and all this great efficiency, I think now is the time to really jump and really take a steep drive forward, not only in productivity, but on the environmental side. I think we can really start to do a much better job placing our nutrients, figuring out where they are. And I think as researchers and folks that work in agriculture that support us, to me, there couldn't be a better time. Any idea is now possible. All the things that we thought were impossible 20 years ago, I think within the next 5 years are very possible, whether it's from variable rate to seeing our fields and figuring out what's out there, measuring soil health. So to me, it's a great time to be optimistic. I think it's a great time to be in ag. And my daughter wants to come home and farm. And I think 10 years ago, I was like, we pushed her away from farming, right? We didn't do the 4-H and stuff. So I was like, it's probably not a good spot for her. And now I'm like, all right, what are you going to study? We doing ag econ? We doing agronomy? Where are you getting your MBA? All those kinds of things that I think are really exciting, and she's really excited for it as well, which is I think it's a great opportunity.
Yeah, I want to agree with Trey on that, and take it to the next level. 3 of my 4 sons are involved in the operation, but people are your biggest asset. The equipment and everything, the technology that we have, what's coming, the laser technology for disease control crops, for instance. Thinking about the fact that it might not be the fungicide that you apply, it might be the carrier is having some kind of physiological effect. Jill Clapperton talks about that. But just basically surrounding yourself with really good people who think differently than you do and who you can trust to give you an honest opinion. Not necessarily your employee, but a group of peers that honestly tell you what they think. And that it's a different perspective than perhaps what you have, because when we do back, look at all the that's where we get in trouble. No. And I think that that's going to be the most important thing going forward, because yes, agriculture is exciting. Agriculture planning in particular is amazingly exciting, because there are so many opportunities here that we have that other places in this country don't have.
All right. Thank you so much. If you would join me in thanking our farmers. I guess my one takeaway message for the folks in the room here today, which are primarily what we would describe as the trusted advisors of our farmers, is that, you know, as Trey said, it's kind of hard to be optimistic right now as a row crop farmer in a drought with low commodity prices. But the optimism of farmers literally never ceases to amaze me. And so making sure that you, as those technical assistance providers, are supporting farmers, are giving them those new ideas, encouraging them to try things, letting them know about the incentives available. They want information from you, is what we have found through research. And so you're really the best people to help them through these times. And so with that, while we're getting our next presentation teed up, I'm going to ask Jamie Kimball to come up and deliver our next sponsor spotlight on behalf of Willard Agri-Service.
Thank you. I'm Jamie Kimball, representing Willard Agri-Service. I come from a family farm in Queen Anne County, Maryland. Main reason I came to Willard Agri-Service because they are an independent ag retailer that's family owned as well. I can pick the phone up right now and call any of the members of the family. It's that feel good place to work. Willard Agri-Service is very excited to sponsor this event once again. It's fun to come here and listen to the farm panel. Willard Agri-Service has been in the plant nutrition space for 55 years now, you know, and also in the 4R practices. And it's where we kind of adapted what we call our farm performance system. And we have that logo up here now. We really branded this back last year in 2023. Main thing is what we need to do is have good trusted advisors, farm performance advisors. So really hone in and listen to our growers and have them tell us what they need and the new challenges they face. I mean, our job is to solve problems, help them increase their profits, and do that in whatever way we can, whether it's logistical services, where they can use some of our plant technologies at our locations, whether it's agronomic decision support systems. The bottom line is that it's our growers that. Bottom line is we need to listen to our growers to really understand, like they said, we got to bring them solutions at $4 corn and $7 corn, and really hone in on those strategies to really make them profitable. Because in the end, their profitability is our profitability. So thank you once again.
Thanks, Jamie. All right. And I apologize. I do unfortunately have to leave you, although I would much rather stay here. I'm taking my 2-year-old who has a broken leg back to work with the surgeon today. So I'm sure you'll have more fun than I will. But our next speaker is going to be Dr. Jared Miller from the University of Delaware. He's the assistant professor and extension specialist in agronomy. And as a Delmarva local, he works a lot on resilience in our grain production system. So please welcome Jared.
Thank you. So I'm going to go over a research project that's been funded by Maryland grain producers, looking at grid soil sampling and a little bit of variable rate application of our research fields. And just in case you're not familiar with the different types of sampling at this point, we are. Most of the types of soil sampling, the original method we use is you do it by field, although in some cases people seem to have to be forced to do any kind of soil sampling at all, to do any kind of fertility recommendations. But you go across this field, mix samples together, and you have one to represent the whole field. And researchers in the 80s started wondering if they could figure out more of the variability in the field. So, of course, being an academic researcher and wanting to overdo things, you think, well, if I could measure every single point in this field, I could really map out nutrients. And they kind of overdid it, and then came up with grid sampling, which Josh discussed a little bit. And after they did a little grid sampling, they started thinking further, well, maybe we do it by zone instead. We can look at zones in the field and look at it towards what's the best yielding parts of the field, and then you end up taking less samples that way. So intensive grid sampling would have been the earliest method. The grid size we'll talk about it. The grid size and sampling method is important, especially when you talk about cost and time fall, which go from taking one sample to a field to, in our case, up to 400 across our research field. When you get into management zones, you start to look at it as towards, well, where's my best yielding parts of the field, and can I address the what's building in that way? So you end up taking less samples. So if you look at these zones that are right here, these two yellow ones are zone 2, the orange is zone 3, and the green is zone 1. You're actually only taking 3 samples in that field. The zone 2 is considered to have the same yield productivity, so you sample the fertility based on that, and you apply based on that. So you go from maybe taking 100 samples in the field or 50 samples in the field down to 3. The problem with the difference between these is, in grid sampling, it's the easiest one to think about because up front, you just walk across that field, you put down a grid, and you take soil samples. All your thinking is on the back end. How do you interpolate the map? How do you make fertility recommendations based on the map? For management zones, you got to think up front. You have to create the zones. So all your work and brain power for management is done up front, and then you take your soil samples afterwards. So grid sampling density, there's been a lot of work done on this over the last 30 years. This is from University of Nebraska publication on the left. It's 42 samples per acre, incredibly dense. This is plot level type data. And on the right is 4 samples per acre, which is about as dense as Josh was mentioning, a quarter acre. So it's pretty dense. You can see on the left how much more detail you get from an intense sampling. There is a question of how much detail do you need? How can we vary our rates across the field? Can you actually dial in your equipment to the density that you put the samples in? So if you undersample, you're going to miss variation in the field. If you oversample, you spend too much money. A lot of the recommendations from the past would have been maximum recommendation is 2.5 acres for 1 sample, but most of the university recommendations are at least 1, and as Josh said, you got to get down to a quarter acre, and you'll see it in our data that the denser it is, the better the map is, but the cost skyrockets at that point. Just as an example of what grid maps could be good for is this is some of the data we'll show you. What you're looking at is pH there across our research farm, and grid samples are good at finding management, past management. With zones, if you understand your topography or your soil type or any of the other factors that go in the field, you can create those zones with soil sampling. What grid sampling can be good is finding out who did what in past field. In this case, you can see if we have 90 by 90 foot plots where we do irrigation, this is a pH difference we had in our field because of the research we've done on this research farm. So one of the things that we wanted to grid sample for was to figure out what have we done in our field in the past, because for all of our research to work, we actually have to understand not just the soil variability, but what have we done with these plots that might affect our research. So this is the biggest benefit of a grid map, is to find some of these past management issues where the zone sampling can still find the yield limiting zones. If you're going to do zone sampling, you have to consider the layers that you're going to look at. So this is that upfront management. This is the upfront brain power that you need. You can use your grid map. If you do a grid map once, you can then use that to create something. So that can be a benefit. If you've done a grid map, and it wasn't helpful with variable rate application, you can still use that to say, well, at least now I know where the high and low phosphorus parts of my field are, and I can start to address it that way. Soil survey, topography. Soil survey would have been an original type layer we would use. You'll see I'll have an EC map in here. We're talking about the dual M and also the Ferris earlier, making these maps of soil type and be a lot more specific. Your yield maps are very important because the yield should be the number one thing that you want to predict where your zone will be. But because you have variation in crops in those fields over time, and you also have various yields due to weather, you have to have several years. They recommend at least 3, but up to 5 years, just so you know where these high and low good parts of the field. Because if you're in Delaware, that sand hill becomes the best part of the field when you constantly have 4-inch downpour rain. And so it varies every single field. This is variable rate map that came up for the research farm. So when you look at, the only reason to do any kind of precision soil sampling should be to do some kind of variable rate application. Otherwise, you spend a lot of money to get the fact of the field which you're not going to use. So this map line rate that we did across the field, so we've got up to, I think, over potentially 10 tons of some of these areas are very acidic, down to less than 1 ton of the line that's recommended. All of your your variable rate application, for the most part, what they talk about is phosphorus, potassium, and lime. So with your lime, you're getting calcium, and we talk about macronutrients. For the most part, you shouldn't be considering doing any kind of micronutrient variability application out there. And for the most part, no one does that because it's additional cost per sample. We have micronutrient maps because we were a grant funded project, and we will pay more money to get that data. For the most part, you're just looking at that. Nitrogen can be variable rate applied as a macronutrient, and some people have tried it with different zone management methods on topography, but for the most part, because nitrogen is so hard to track, changes so quickly in the environment, we usually look at it. Some of the better methods have been applications through sensors. Such as this, the project that we are doing in this area. Dr. Nicole Fiorellino speaking after me, is a project run by her. NRCS is sponsoring CIG, where we're looking at using sensors to make these nitrogen-based applications, which started out of Oklahoma State and places like that. And we're looking at right here, and even Josh did some of that work, I guess, 15 years ago. So this is the project we ended up doing. This is our irrigation research farm. We wanted to do grid sampling. We were working with some other people on doing variable rate lime application, but we decided that if we were going to screw up a field, that we should screw up our own field first. So we did quarter-acre grid sampling simply because what James does out here is he does variable rate irrigation, and he does it in 90 by 90 foot blocks. He's done it on 60-foot blocks, but he found that it bleed over between the microenvironment with moisture, so we got it on 90 foot. So we ended up doing about quarter-acre grid sampling across here. They don't line up. This is our variable rate linear. This is the north side of a variable rate pivot and the south side of a variable rate pivot. We also have subsurface drip irrigation over here. I haven't added that. So we sampled in April 2022, and after we did a variable rate application in 23, we came back in 2024 this year and sampled again to see how well we did. And the Delaware Soybean Board actually paid to send off our top samples to Texas. We do have Texas on the side. So here are some potential sample density. This is sample density that we did. It's a quarter-acre grid. I went across through here a couple years ago when we first did it, and I deleted out samples to see how poorly did these maps start to perform if you went to lower resolution. So here's how many points you have on a quarter acre. I think across both farms, we were looking at over 400 samples across this whole place. So we're talking about $10 a sample or $20 a sample across both. It's up there. When you go down to 3/4 acre, which is the easiest way to read out points, so they're not even in favor of the quarter to one, but you can see you really reduce it, get to 1.5, it's barely a whole lot of effort. When you get to 3 acres, you're not taking many more samples at all. But the differences really come out when you look at the resolution of the maps that come out of this. So here's your soil pH at a quarter acre. You can see here is our past field variability, here's our soil variability, here's field management. As you go down to 3/4 acres, some of the patterns are there, but you start to lose. We've already lost some of our field management. If you get to 1.5 and on up to 3 acres, and you really don't know where that variability in your soil pH was, and all of the nutrients are the same way. So some of the comparisons when you average across the field, when we look at average samples, are pretty similar. But when you start to look at what's the quality of this map, you might save money and time by going 1.5 to 2.5 to 3 acres, but you're not really getting a quality map at that point. In terms of if you want to address this, and this is some major variability, you're talking stuff that's less than 5 and things that are up to 6.5 and 7. So a lot of field variability out there. And if you're going to do variable rate application at this point, you're not really going to do it and improve it very well. This is a different study. I'm just using it as base numbers as you go across here to look at the data that we got out of the field. This is another Maryland Grant Producers sponsored project, also done with Nicole, where we looked at our variety trials over a 3-year period from the coastal plain all the way out to the Keys, Hill, and the Ridge and Valley, so we can get a base idea of what the nutrient uptake was in these corns compared to yield, and also what our soil levels were. So I just used these tables as we go across here to look at some of the values that we have from our samples. This is a 1/4 acre grid map of cation exchange capacity, and all my maps you see on the left is April 2022, and then again in February 2024, and these are just some of the patterns you see, so low CPC up to high CPC. Across the region, from the coastal plain to Keedysville, we had CECs across the Mid-Atlantic ranging from about 4 up to 12. So that might not sound like a lot to some people, but if your CEC goes from 2 to 4, as we get in our Delaware fields, we double the nutrient holding capacity. So it is important, although those numbers might appear small, if your CEC goes from 3 to 9, which it does in some of our fields, we triple the nutrient holding capacity across that field. So minor differences in our region because our CEC is so low. So here is your low CEC. If you look across both years, the patterns in the field don't match up very well. You kind of expect that. Your soil test CEC is a very quickly done test. It's a cheaply done test. It's a sum of patterns, which means when they extract out your calcium, magnesium, potassium, sodium, they add it up and say, well, this is your CEC. It's not the most accurate method. So if you look at a grid sample map of CEC, it's already going to have errors. It's going to have errors in how you sample it. It's going to have errors in the lab, and it's just going to have errors in the method. So I would never expect CEC map to match up very well. And if you go across here and variable rate apply nutrients, and they're based on a sum of base patterns, the actual values, then over time you CEC match. So if you get a lot of variability in that, send it to different labs, and everyone has different employees working there at different times. This is something that could vary a lot, so I wouldn't expect it to match up. So our CECs at Warrington, though, as you'll notice, they run from about, see it on there, it's 3 to 9. So at Warrington itself, we're running from CECs are as low as 3 in this area up to 9, and all the 9s, of course, are on the edge where we don't do any research. So this is a very poor field for us to do any kind of nutrient uptake research unless you want to start plants, which is typically what happens. So we have a lot of variability in this field, and so Warrington, you're going to see a lot of this data actually matches the variability across the entire Mid-Atlantic. This field used to be part of an NRCS mapping exercise because it was one of the most messed up fields that could send all the employees down to, just in terms of soil type. So this is just a point of how a grid sampling map is useful. And grid sampling map for me is showing you a lot of variability across this field, but all these other different layers that go into zone maps also give you information. So up here we have our CEC map, and this showed us some patterns we've seen with nutrients across the field. This is a drone image of soybean on the north side of our pivot, and if you see these patterns right here, these patterns go in aerial photos back through time. Here's an EC map done by Barris, which in this case is mapping texture. These are old sand hills because we're a coastal plain field. We have old sand dunes possibly running across here. This is nematode damage. It shows up in corn at the end of the season. So even though we have a 1/4 acre grid sample at CEC, and we can start to go across here and base applications on this, we have other information over the years that tells us that we still didn't get this level of detail. I have a soil texture map that doesn't pick up these sand. Takes it up a little bit because between those points it just wasn't dense enough to see this little bit of data across here. So although the grids are helpful and the grids have shown me things, I was originally a soil mapper, so I look at it. I was called in that kind of type of soil science a lumper, not a splitter, which means instead of having 6 different feed horizons, I combine them together because I don't see the effort in splitting everything out. But when you look at the grid match has shown me a lot about our research field, but we have so much other data over the years that shows us variability in the field that it didn't pick up. So take all these maps, although some of them might be interesting and show you some of the field variability, realize that there's a lot of other data layers out there that help you find yield limiting zones. And in this case, it doesn't matter how much potassium I put out there for soybeans. Our yield limiting factor is nematodes, and we have to correct that first. And then after that, it's sandy, it's hydrogen water holding capacity. So all of those things are going to reduce my ability to improve yield, and then that potassium I apply in there isn't going to matter anyway. So you do have to think about these things if you can get this type of. These are our 1/4 acre organic matter maps. You shouldn't have to map organic matter every single time. It's not going to change very much. No matter how much carbon sequestration you try, you're not going to build it or change it that much over time unless somebody puts 20 tons of compost down in the field. However, it's pretty common to get organic matter every time you sample, so you'll probably be able to look at that. The patterns across the field are similar because values change and we wouldn't expect them to change. And in this case, the one texture map that we do have for the North Pivot, all these orange areas are high clay, and to expect in the coastal plain, when you have a lot of variability in clay, that's what organic matter is. So for the most part across here, wherever you have more clay, you also end up seeing sulfur on aluminum is. Wherever we have more clay across this field, we have more organic matter. So your organic matter map, even though we did a texture map, coastal plain soil might actually be a good proxy for your soil texture as well. This is a quarter-acre sulfur map. So sulfur is pain. The soil tests for sulfur aren't going to be very accurate because sulfur is an anion. It's going to move through your soil. It's going to leach. It mineralizes from organic matter. It's going to change through the season somewhat. The same thing as nitrogen does. So you should never take your soil test sulfur as something as a solid measurement to base a recommendation off of. You have to do several years of your own knowledge of the field and tissue testing to really figure out what's going on with sulfur. The benefit of having this map to us is because we try to do sulfur research and try to get a response and have it. Is now we know at least across Warrington, over the 2 years, April 22 and February 2024, the absolute values were different. Because you had April numbers and then February, we have the mineralization of sulfur, you have cold winters, you have crop uptake, all these things are going to change sulfur over time in this field compared to something like micronutrient. But at least we know what you'd expect when you have more organic matter, when you have more clay, so you have more sulfur in the coastal plain field. And the high spots in the field over both years remain the same. Assuming you get borons, that wasn't the case. So for us, in terms of doing sulfur research on this field, we now know where our higher parts are. If we're going to do different studies, or we're going to worry about where our sulfur sources might mess up, we're going to block our studies across this field. A good idea. And I also have a better idea of sulfur across this field over time. Wherever we have sulfur, one year we should have it the next. And it's only 2 years. I wouldn't expect it to change much. So across the area, we had 8 to 59 ppm sulfur across the Mid-Atlantic. But again, those numbers can change so much, I don't think I would trust it. This is a comparison over 2 years, how well the soil test lab do. It's not a great line. You'll see it gets even worse with boron, but I wouldn't expect sulfur between 2 different sampling points to match up. Sometimes the best sulfur number I've gotten from the crop uptake is when I take it mid-season, because the sulfur that was available in July seemed to predict better what the crop uptake would be. These are the zinc maps. So all the micronutrient maps, they shouldn't change. You shouldn't have a lot of variability. When I compare the absolute values, the UV soil testing lab did a really good job between 2 different sampling dates, potentially different points, and having very close ppm. Also considering that we're taking 4-wheelers out to the middle of the spot in the field using GPS and sampling the soil. So there's going to be some variability in the data that comes out. So the trends, of course, we have higher zinc on the southern half of the pivot, but over time we did get some variability in the absolute values. But the numbers that are over here that you can't read are going from 2, 3, 3.3, 3.4, 3.6. So if you scale this out, the differences across the aren't a lot. The zinc is pretty low in this field, but it's still enough for crop production. Across the region, when we did that study a couple years ago, we went from 0.76, which was low, up to ppm 18. This spot here, I think it's ppm of about 24, and this is where vehicles are parked. There might be great dust there. I don't know what's coming off this edge. They've had more problems in the past, so that zinc is probably correlated to management than it is to anything natural in the soil. But what's interesting across here, here's some of your lower CPC zones. When you start to look at iron, aluminum, manganese, is when you start to see some of the trends in the field and how different they are. The zinc, over the 2 different sampling times, had a better relationship. However, if you've done enough regressions like this, you know that when you have 5 points out here and a bunch clustered down here, these actually end up making this look better. If I deleted these points that are very high and just looked at this clustered relationship, probably not as good over time. But we wouldn't expect things to change a lot, and it's still within a range of sampling. The iron map, I've kind of outlined where the high iron is because there's a pattern with that when you look at, we want to eventually look at the elevation across this field, and potentially where sand is laying down. So these maps are interesting to me because, yeah, I'm a soil scientist. It's not always as much fertility. because I like soil mass and how things form, but also figuring out how this got here at some point, whether it was man-made, whether it was natural, and then how do we address it when we're looking at, we've got iron moving across these fields and affect phosphorus absorption, and iron and aluminum in terms of pH. More iron in the soil and you have lower pH, you might have more toxic effects. So there's different things that you can think about in terms of plant productivity. So the high iron is across here. Across the region, our irons from the coastal plain to Keysville was 63 to 230 ppm. At Warrington, our iron went from 61 to 271. So this field itself matches the variability in iron across the entire mid-Atlantic. We have a lot of variability in iron availability. We typically don't have any kind of iron issues here. You can raise your pH up close to 7 and still not see any kind of iron problems, any kind of iron deficiencies. Unlike if you read a lot of literature from the Dakotas, they end up having a lot of iron deficiency in their beans and corrosis from that we don't see here. So although iron could be a problem, we typically don't see it here in the region, but still the map is interesting when you look at where the manganese was. It's ended up being opposite. In a coastal plain field in Sussex County, Delaware, here, same outline for iron, here's your high manganese. And it drops as we go across the field. So again, for me, in terms of fertility, we can do some different studies on availability and pH, but just seeing how much this manganese varied across the field. If we want to do manganese studies now, and we have nothing compared to Keysville, this is 262 parts per million down here. They have a lot of iron. Clarksville was definitely out in Appalachia. They have a lot of manganese in their soils out there. And it does affect uptake. In a study we did with grain producers, all the fields that have higher manganese had higher manganese in the tissue. We do have manganese deficiencies a lot in Delaware. It's probably the #1 deficiency in micronutrient we see. Zinc up around Middletown, but manganese in certain soil types. And Fallsington, if that soil is one typically in Delaware that has manganese deficiencies, and it gets induced a lot when the pH goes up too high. Because as your pH goes up, more manganese absorbs to the soil, and then it's not plant available. Anything that's bonded to the soil can't be taken up by the plant material. So you have to have a more acidic soil pH to have manganese availability. But what's interesting in this fact to me, not knowing it before, is you see we have this high iron that goes across here that's the opposite of manganese. So is that effect of how this coastal soil was laid down, the source of the material, or is there some kind of interaction? Because this is still an extraction. It's an acid extraction, pulling manganese and iron out of the soil. Is there some kind of interaction between iron and manganese in the soil that also affects availability? Oh, left. So the manganese has a much better relationship over time. Copper map makes no sense. I've got nothing to say about this. I have no idea what's going on with copper. I don't know if it's natural or man-made. I don't know what somebody might have done here over the last 50 years. I have no idea what it looks like. I was very surprised, but it happened 2 times in a row. Over both sampling times. We have very low copper here. We have much higher copper across this part of the field. It's on the south side of the pivot, but it could be natural. It could be something that happened 50 years ago because of someone else's application of it. This is the boron map. So the first year we saw it, John got excited because down here, where this cuts across on this side of the pivot, James dug a ditch out there for me. And we find that was 10 years ago where there's some kind of soil effect of boron because boron's a pain. It's like sulfur, it's an anion, it leaks out of soil. The more organic matter you have, the more availability it should be. But when we came back in 2024, the map didn't match at all. So February 24, April 2022, can't get the same amount of boron. Boron is a pain. Boron's a pain to research because we don't see any effects out of it. We definitely need it. We definitely see where plants need it available, but it doesn't always come up. And here's your relationship, which you never test. You should never trust the soil test boron that comes out of the lab. It doesn't probably mean much. You need a tissue test, and that should feel good. The aluminum map, again, as a soil scientist, is one of the more interesting ones to me. So we have low aluminum here with high clay. A lot of our high aluminum is associated with clay pockets across this field. Again, this could affect phosphorus absorption. It also affects our buffer pH and how we might line these soils. And what we've noticed across this field is this might be an old sand dune that goes across here. So here's your high iron, but low manganese. And also we have low aluminum across these zones here. So these nutrient maps or element maps across here are giving us a better idea of, for me as an old soil scientist, how this field might form. The availability of these low spots in the field have more clay, but also more aluminum. So that's an weathering effect. And can I start to look at plant growth? So we'll get into the variable rate application with what time we have left. We got a new dry spreader, variable rate conveyor belt. We have these spinners that can control how far out we spread it, and we can vary the rate of these dry materials that go across the field. So the first thing that we looked at is we wanted to fix the lime across. We want to fix the pH across this field. This is a research field. We're not doing line studies out here. When you do any kind of variable rate liming or liming at all, you need two pHs. You need your soil pH that tells you what the crop sees. You also need the buffer pH, which tells you how much acidity is bonded to that soil. So if you look at these two zones here, we have a low pH. We also have more acidity on the soil. It combines into making this map. So you can see that we need a higher lime rate where we have both a low soil pH and a low buffer pH. But where our buffer pH is a little more moderated, we end up adding not as much lime. And over here, where our pH is higher, and it says that it's not as well buffered, you really lower the lime rate. So that's how those two pHs combine across the field. Question: how well did we do? A simplified map by putting it into anything less than 5.5, anything less than 6, and this is 6 to 6.5. So we want to be in this blue zone right here. Here's 2022. A lot of acidity across this field. A lot of stuff less than 5.5. We should expect some aluminum toxicity. A lot of lower yields just due to not managing the pH across this field. This is one of the reasons we want to do this. And you can see that we did a pretty good job on the south pivot, getting that pH in that range that we wanted, about 6.2, so between 6 and 6.5. But right in here, didn't raise much at all. So what's interesting is that it's actually an area of the field where the buffer test, which is Adams-Evans, D, which you're going to probably phase out, as Adams-Evans said, it's not well buffered, don't add more lime, and we didn't, and we ended up not raising the pH up as much as we wanted to. So it's possible, in this case, we could have done the calculation wrong, did all this. Possible that buffer pH in this area was underestimated, the amount of lime. So we wouldn't have known this before without this application across this field. What it really did a good job of, and I'm not a proponent of having even base saturation, but we did do a better job of getting the base saturation across the field. But this is also going to include potassium application. One thing that you can do once you have this manganese map, though, manganese, we do have an availability index based on pH. And you see right here, if it's in gray, it means you should have no issues with manganese availability for crop uptake. Anything in yellow means watching, anything in red means you have a problem with definitely have manganese issues. So here's our low manganese, here's our high. In 2022, with all those acidic soil pHs, we didn't have any problems across the field. Once we lined it, when you use this index, it comes in a QP recommendation. They have ones like this across the country. You can see that we now know at least, we raised the pH where we wanted it, which I'd be a proponent of. Get your pH where you want it, but we now know that we should potentially have the watch manganese here, and this is where you might do a foliar application. But this is where you might bring up the manganese concentration. But we shouldn't expect it across the field. So another thing we can do is we can do more studies on manganese on this part of the field. But something like this, a map like this, you can use to then decide, instead of a blanket application of foliar manganese, you could end up trying to concentrate where you might see the problem. And that would be a benefit of that map. Potassium application, we're at the bottom of the field. We did pretty good. This is what research does. So whatever we were doing over time, we really lost the amount of potassium needed up here. So here's the recommendations are pretty low, close to zero at the bottom of the field. Some higher rates across it. We end up matching CBC. While we fixed some of it, we didn't fix all of it. We didn't really do an even application. And some of this might come from how well the soils hold potassium. If you come over here to the SDI, we've done some studies here. This zone right here has, across the middle, has CBCs less than 4. The potassium out of that field with just regular broadcast potassium, it's not holding as much. So lower CEC potassium is going to reach out of the field. It's not going to hold it. We've done some studies in here to try to see if uptake mattered or not. With potassium, we've done applications with and without A within these different zones, and it didn't matter. Because Delaware has another issue. We have potassium in our minerals that soil tests don't pick up. So we have 24 ppm to 50 ppm potassium in this field. We should have deficiencies. Adding K didn't matter. The problem was across the zone that's the water hold. So again, you can use a variable rate K map, but if you don't understand that your limiting factor is moisture and water holding, and as Josh was talking about, maybe where the roots can go because the soil just can't support it, then your potassium application is waste. So we don't know for sure until we come back and do another application this year. Can we address this? But do you? If you have a low CEC and you apply too much potassium, you might have issues with calcium magnesium uptake, which is another thing we did one of these fields. So you can't just think about, I want to get this field even. You have to think about all the factors that affect nutrient uptake, crop availability, and you probably never get it perfect. But this is some of the things that Josh was talking about. We need to do more research. Understanding the availability of these nutrients and crop uptake across all these different zones. So when you look at grid maps and nutrient cycling, it gets more complicated. One, we can't see all the variability in this field, but also, do we understand all the relationships across these soil types, across environment, across topography, that would help us do a better job of variable rate application, and not just using a grid map by itself. And we don't, and we might never get there because some of this stuff is so complicated. Variable rate phosphorus, don't do a lot of phosphorus research because our speed is so high. I leave all that stuff to everybody else. No one knows it, so don't work on phosphorus. He did a variable rate application. Didn't put enough out. Barely moved the dial. He brought in Amy Schober, our nutrient management specialist, to up the rates this year to try to. But we're not trying to even it out because over here we're talking stuff that's over 150, I think up to 300 FIVs of ppm, and stuff over here that's closer to 20. So we just are trying to use unique recommendations to get this field in a zone where we want it to be. But we didn't add enough. We barely changed the phosphorus across there just because it did the wrong calculation. That's the next thing. This is, it's all right. Got it. This is just what the field looks like this year. Maryland grain producers sponsoring a biological study on this side, and we have some strict intercropping on the other side. And we're taking tissue samples throughout here in zones that are more controlled to try to see how we'll match up with the grid sample. So you can see how all this research this is not just biologicals. We're putting nitrogen out there to make sure they work. You can see what happens when you don't put nitrogen this year out in these fields. They all dry down a lot of it, even though it's irrigated. So this is why we have to do so much work in this field, because there's so much different studies going on out here to make sure that what we're looking at matches up with some of the nutrient uptake we see across the field to try to do it. So I think we're out of time, but if you have any questions.
You must have done a good job.
Yeah, thanks very much.
Anytime.
Sorry, I'm short. Thank you so much, Jared. We truly appreciate you and your research and bringing it here to us. Sorry y'all are stuck with me now. I like to keep things spicy. I was a state FFA officer, so we moved a lot. So before we hear from our sponsor highlight, turn to your partner and play rock, paper, scissors really fast. Alright, who won? Raise your hand if you won. Alright, I have a 3-year-old and he wins every time. Okay, so Before we begin our next presentation, I want to invite Alex Eccles up with the Keith Campbell Foundation. The Keith Campbell Foundation is also a sponsor of the Mid-Atlantic 4Rs and also funds a number of our projects, currently our 30 voucher program. So please give Alex some time.
Thank you very much, and I won't take much time because I've been in this room a long time. Carolyn Harper and I are here with the Campbell Foundation. Campbell Foundation's very different than most of the foundations that are in the environmental and conservation business. We view farmers as the essential partner. We don't want to tell anybody what to do. We don't want to push more waves on anybody. Our business is water quality. There are two things that we try to advance. Water quality, yep. We don't think we can get there without farm economic viability. And so that's a very important component of what we are. I'm 68 years old, I have to think of it. I'm a sophomore. Everybody knows what the origin of the word sophomore is from Greek. It's informed fool. I'm not sure I'm very well informed, but I'm early on in the learning process, even though I've been at this for almost 50 years. We're here because we need to hear from you. We invest in how to advance farm conservation. We believe that to do that, you need to engage the economics. If it doesn't pencil out, it doesn't stand. And we're very serious. Please don't ever hesitate to be in touch with either one of us. If there's real work you want done, call Caroline. If you want to talk about hunting or fishing or beer drinking or something, no, seriously, we need to hear from you because you know how your operation works. We're not trying to tell you that. Thank you.
Alex. For our last speaker today, Dr. Nicole Fiorellino is presenting her research entitled Updating Fertilizer Recommendations for High Management Wheat in Maryland. As the Assistant Professor and Extension Specialist in Agronomy with the University of Maryland, Nicole works hard on research needs that the Maryland grain farmers need. Please welcome Nicole to wrap up our event today.
Thank you. Oh gosh, this is so fuzzy. All right, we're gonna go with it. I can't stand still. Sorry. Thanks, Janelle, and thanks for having me. Really excited to be part of this event again, and share some information with everyone. So, Janelle and Lindsay asked me for a title of a presentation a little while ago. So I gave them this. Well, they kind of requested that I talk about this topic. We're going to get to it, I promise. But we're going to take a little bit of a journey to get to talking about my results, mostly because I only have 1 year's worth of data. All right, that's it. I don't have a lot of exciting stuff to talk about. All right, so we're going to go on a little bit of journey, and I thought it was really appropriate for this audience. We've heard this said kind of multiple times today. You guys are, the trusted advisors, the trusted consultants. That's not to say that there's not farmers here in the room, but for the most part, you guys are the advisors that our farmers are going to and seeking information. And I think it's really important to share some background information and some insights about how you guys, us too, as university folks, extension folks, are providing recommendations. So bear with me because I realize on the last talk of the day, we're gonna talk about statistics. All right, so just wrap your brain around it, buckle up, let's just do it all together. All right, and I'm doing this in a very practical way. I think it's really important that we're understanding how we're analyzing data. And Josh really kind of hit on it this morning. I was joking that we need to communicate a little bit better when we're on the same program and we're talking about very same topics. So we have parallel messages. But a lot of times when we're looking at data, there's multiple lenses that we're looking through this information. All right, and Josh kind of hit the three on the head, right? Trying to maximize yield from an economic lens, maximizing profitability, and then in our region, it's always the environmental lens, right? So 3 different people can look at the same data that I'm about to show and make different interpretations. Whatever, we're all different, and that's great. That's what makes the world go round. But it's really important to understand that there really are fundamental principles that we all need to be on the same page about, and there's data that we can throw out because it's junk and it doesn't mean anything. Okay? No amount of interpretation is going to change that. So buckle up. We're going to talk a little bit about statistics, and I promise I am going to show you data, and you get to put to good use what you just learned and critique the data that I'm about to show you. Okay? So let's follow along on this journey. All right, so talking about nutrient recommendations, Josh set me up. He gave a lot of the background information that I was going to talk about. Really, the most important point that I want to hit home is that every state has fertilizer recommendations, right? I did a little Googling one day. I was kind of pulling up different examples here, and I was checking the dates on them, all right? A lot of the recommendations in other states, same as in Maryland, are old, all right? Why? Josh kind of hit the nail on the head, and I purposefully asked him that question that I asked him. People are not performing this research anymore. It's expensive. We're going to get to that, okay? But also, generally, our systems are kind of stable, right? What is me running nitrogen rate trials over and over and over again going to tell you, right? We're growing corn. Corn is corn. Really, our systems, our rotations, there's not much changing. So I think we have this idea, we in academia, these recommendations are generally stable. In pretty much every other state besides Maryland, these recommendations are just that. They're just recommendations, right? Do you think people in other states really have memorized what the university recommends on nitrogen for corn? No, because they just do whatever they want, right? But we are in a region and we are in a state where those recommendations get turned into regulatory tools, okay? And so you would say, well, it's really important that we update these in Maryland, Nicole. But as Josh alluded to, this kind of research gets political, and some people like to take a step back, all right? So just kind of setting the stage as we're talking about what recommendations are. Josh said it's really expensive to do this kind of research, and it is, because we want to collect data so that we can make graphs like this, and that, and that. That's a beautiful one. That's a real one. I took that from a publication. Josh gave me that one. I mean, imagine how much sampling and how much work went into putting all of those data points together, right? That's a lot of data points. Yeah, okay. It's a lot of data, but it's important, and we can draw these really nice lines. This one's actually super impressive. Really be able to draw those really. We want this kind of data in Maryland if we are updating recommendations, because our recommendations become regulatory. They don't sit static in a book that nobody ever looks at again, all right? Do you really want our fertilizer recommendations to be based on a straight line that's drawn based on one field's worth of data that looks like this? No, right? We want to have confidence. We want to look at a graph that looks something like this and be like, dang, I collected a lot of data points. I feel confident that it was done the right way, and I feel confident in that recommendation, all right? So I know Josh verbally said, we don't need to collect more data, we need to collect better data, but I'm going to say we also need to collect more data, okay? So a caveat to what Josh said. We do need to collect more data. Okay, so let's talk a little bit about statistics. Why is this important? Why do we need to collect more data? Why do we need to make sure we are collecting sound data? All right, hypothetical example here. Let's say we want to test corn yield response to some kind of a yield boosting treatment product, whatever you want to call it. This is your field. You go, yeah, it's a really nice square field. Let's take that field and let's split it in half. And we're going to do our farmer standard practice on one side, and we're going to apply our treatment on the other side. We're going to compare the yield on the two halves of these fields, and let's see which one's better. All right. So apply our treatment on the one half. And then we measured our yield. This is what we ended up with. This half of the field yielded 132 bushels per acre, and the control 126. Conclusion: the treatment increased yield. How do you feel about that? You sure about that? Really? Yes, I knew there was going to be some young people in this audience that were going to laugh at my meetings. Thank you, Emily. Are you so sure about that you would bet money on it? I'm not a betting person. I'm so risk averse. But I mean, really, just by splitting one field in half, that's almost as bad as that graph of wheat response data I showed you, right? It's like those 7 points. Did you really bet on that? I wouldn't. Especially if you go back to that same field and you overlay a map that's looking at slope here. All right, so let's put our squares back up again. All right, you just happen to put your control on the half of the field where you got all this slope. All right, Jared, Josh just talked about when you see slopes in the field, you know those parts of the field don't perform well. Did you set that control up for failure? Conversely, did you set that treatment up for success? Maybe you didn't do it on purpose, right? Maybe it just happened. You decided to split your field and you just happened to put your treatment in the better half of the field. Are you really sure that that yield benefit was actually from the treatment you applied? Really? You're gonna stand up and say you did that? I don't know, not me. So let's try this again. Now we know that there is some variability in the field. Besides the fact, when we just have 2 data points, we can't use statistics. I know statistics seems like a black box. It's actually a really useful tool to give us more confidence in that response. All right? So you say, Nicole, I'll buy in. Instead of just splitting the field in half, let's split this field into a number of strips. All right? And we're going to use the same area of that field. We're going to split the field up. We're going to put 7 strips of the control, 7 strips of the treatment, so you can make your averages, Nicole, and so you can run your statistics, right? Sounds like a conversation I have with people doing my on trials. We'll humor you, Nicole. We'll do it your way, all right? Is this really that much better? All right, if our treatment, our product that we think works is in the red and our control is in the yellow, have we really done much better? We still put most of the spots on the side of the field that we know is better, right? Overlay that soil map again. We still put all those treatments over here where it's better. So we knew there was variability in the field, and we took like a baby step to try to correct for it and to try to identify it, but we didn't really do that much better, right? We replicated, but we did not randomize. Okay, let's take a look at that data then. When we harvest each one of those strips and we just simply plot the yield that we get, so the red are those treatment strips, the yellow are those control. This is just straight corn yield on the y-axis. All right, what does that look like to you? As Gene would say, a bunch of random numbers, random number generator. All right, so the treatment yielded 130 bushels, so the control yielded 120. You might be tempted to say that the 130 is better than 120. Let's look at this data in a different way. This data, averaged together, created the height of these bars, and these little tiny error bars show that variability, all right? So when we stand up in the front of the room and we show you these p-values and we tell you that there's significant differences, we're really just analyzing the differences in the overlap of these error bars. Let me circle that for you, all right? These error bars are overlapping with one another. It's really hard to see that there's a difference here. Just the same as it's hard to see it here, right? They just kind of look like random numbers. You squint your eyes and turn your head to the side, you're still like, well, I think the yellow kind of looks similar to the red or the pink, okay? And so we're using statistics as a tool to definitively tell us, you know, is there actually a difference there, all right? So I took these values, ran them through our statistical software, spit out our p-value. What does that mean to you? That means that there is a 66% chance that the difference is due to treatment. I'm not a betting person, but I would bet on that, all right? We knew there was variability in that field. We tried to correct for it, but we really didn't do a good job. So we weren't able to see that this difference here was actually due to this treatment. I have questions. I don't believe it. I'm not convinced, okay? We also had a pretty high CV. This is a percentage of the coefficient of variability. There was a lot of variability out there. We knew it, we tried to fix for it, but we didn't, and it impacted our ability to find that difference, okay? Now, I'm saying this and it seems very malicious. We knew it was there and we didn't do anything about it. Sometimes we don't know it's there, sometimes we don't do enough to correct for it. Sometimes you just cannot find a significant difference. And I'm going to show you that in some of my data later. All right, it doesn't mean we did anything wrong, it just means we didn't identify and we couldn't account for all of that variability. All right, take 3. We're going to try this one more. Oh, so your results. Would you conclude that your treatment increased your yield? I don't know. Here's another meme for y'all. That's like 60% of the time it works every time. All right? I don't know. I don't buy into that. All right? I'm not sold yet. Let's try this one more time. Same field, control and treatment. This time we're going to do a better job of controlling for, identifying that variability, and trying to do something about it. So we saw the soils map. We know that there are parts of this field that have differences in slope. So this time when we lay out our plots, we're going to put replicates, or we're going to put blocks, all right? We created these 5 different areas, and within each of these plots, I flipped the coin, and I randomly assigned whether it was pink or yellow, all right? This is legitimately how we do it in the field. I have a random number generator on my computer, I throw my treatments in, and it randomly generates the order of them, okay? But we know that there's variability in this field, and across all that variability, we want all of our treatments to be there, okay? We know it's there, we got to account for it in what we're doing. You'll notice, if you're still awake, if you're still with me, I actually have less replication this time. There were 7 reps, or there were 7 whatever strips per treatment in my last example, and I cut it down to 5 here. We're doing a better job of accounting for that variability. Hopefully we don't need as much reps. Let's take a look. All right, when we look at this data again, we are comparing side by side in each one of those 5 blocks, the comparison of those treatments. There's a much clearer pattern here. Wouldn't you agree? Everybody who has their eyes open, who's not actively snoring, right? It's very clear when we look at each one of these blocks that that treatment out-yielded and outperformed that control, right? It's pretty darn obvious. Now when we see the treatment average is 132 and the control average is 125, we might go, there might be a little more to that. When we represent this same data and we make it a tall bar and we look at those tiny errors, that's what we love to see, all right? This is giving me confidence that there truly is a difference between this treatment and control. There was variability there, and we accounted for it. What do statistics tell us? Dang, look at that low p-value, all right? Dang, look at that variability. We're looking for something less than 10%. We got 0.6, that's awesome, all right? There's a greater than 99% chance that the difference that we're seeing here is due to that treatment, due to what we forced to be different. We saw there was variability, we controlled for it, and we accounted for it in our model. We accounted for it with our statistics. Our variability was identified and accounted for. Conclusion, treatment increased yield. Yeah, I buy that. That's what I'm talking about. I would go for that one, all right? All right, so that was an interesting journey. Maybe that was the simplest way statistics has ever been explained, but I think it's really important to have a fundamental understanding of why we do things, why we do research the way that we do, and understanding what these results, what these statistical results mean, all right? Because at the end of the day, I'm not even talking about the next step, which Josh kind of talked about in his presentation. That's the interpretation of this. What are you going to do with this information? I can stand up here and tell you that there's a significant difference all day long. But what does that mean to someone? How do you make a management change? How do you make a decision? How do you as a consultant advise a farmer who's asking you what they should do based on this information? All right. I'm going back so I can show this. Is a 7-bushel increase really worth it? It depends, right? Classic extension answer. It depends. Not only does it depend on the product, it depends on the individual, right? It depends on that individual's operation. The farmer panel sat up here and said, we need economists to help us with this information. 100%. That's my limitation. I'm not an economist. I don't know. I can't tell you that information. But let's take this to the next person to help make the picture, make the story more clear. All right? But we've got to start with this solid foundation. Okay. So I promised that I would come back to recommendations. I promised Janelle that that's what I would talk about, the results of the study that grain producers paid me to do. So let's get back to our recommendations. I talked about how they're expensive. Josh gave a good plug about how everybody should pay the researchers to update these recommendations. And I get this question all the time. Why aren't we working on recommendations? Why aren't we updating them? So, number one, we've started some work on this in corn. And I know that I've told grain producers and I've told people that have asked me, don't ask me to to do corn nitrogen recommendations because I don't think the results are necessarily going to be favorable. However, the purpose of this study that Jared was presenting some of the results on that we're really excited, we're in our final stages, we're kind of wrapping up, was incorporating nitrogen rate trials into our on-farm trials program. Basically, what Josh said, we got the federal government to pay us a lot of money to do nitrogen rate trials because it's expensive. It takes a lot of money to do it, all right? Within this, though, we really wanted to do an evaluation of some of the in-season nitrogen modeling tools that some of you guys in the retailers are, guiding your customers through using. And to be really clear, we at the universities with the PIs on this project, we think that they're really good. Any step forward with precision and making more precise and making better nutrient recommendations is great. Folks are often limited, though, in the state of Maryland because of regulation, right? So we sought out to really head on in a field, strip, compare how do these end models really differ from our university flat rate recommendations. And honestly, we really had no emotional attachment to the results. We truly just wanted to see how they perform. And from my opinion, what has been a really exciting outcome of this study is that the university flat rates and the in-season models, they're really not recommending that much different rates, okay? Which we are hoping to take that forward to folks like MBA to say, hey, there's really no harm in allowing our folks to be able to be more precise, use these nitrogen models, so let them use them, all right? Let's make it easier for them to be able to use them as part of the nutrient management plan. That's really what we're hoping to kind of be our end goal here. So we're working on it. Corn is happening, in progress, we're working on it. I really was a little more interested to jump into wheat, okay? Wheat is definitely a little less political, right? Most of us would agree. I mean, nitrogen in general is political, but wheat is a little less. We have a potential here. We do have folks who are growing wheat in a high management system. They really are looking to push yields. I really just want to make sure that our nitrogen recommendations maybe aren't limiting them, which they're probably not, but hey, let's collect some data. So when we talk about corn and our rotations in our systems, I say, yeah, we're really not changing our rotations. And, that's generally true for corn. But I think when we're looking at a crop like wheat, which is following a cash crop like corn, where we have gotten much more precise, much more dialed in with the nitrogen that we're applying to our corn, there may not be as much residual left over as we're going into the fall, as we're going into planting that small grain, planting that wheat afterwards. So it's not necessarily that we're changing much about how we're growing wheat or what we're doing with wheat, but we've definitely seen changes and definitely seen improvement with how we're managing that nitrogen on the previous crop, which is that corn. All right. Or a lot of people say, like, hey, look, I just don't have as much nitrogen left over at the end of corn. I'd like to put more nitrogen down in the fall. Can I put more nitrogen down? So the farmers are asking about it. Okay. Part of the impetus of why we're doing it. Another major point is that now that we have created the on-farm trials program under my program at Maryland, we now have a vehicle to actually be able to collect this data. All right. I will say I drove my feet on this for a long time because I didn't have the resources. I frankly just didn't have the manpower to be able to collect this kind of data. We're ready, we're willing, so here we go. All right. Skip that. All right, so let's talk about what we started in 2024. This was the first year of our N-rate trials. I decided to tackle this in a different way here. I'm trying to maximize time, trying to do this somewhat as quickly as possible. So we chose to go with kind of a paired approach. So we had two very parallel, similar protocols, but different. We had one protocol that we executed at two of our university research and education centers. This was done on the small plot scale where I had super ultra control to an extent of what happened on these plots. We have professional farm managers that their job is to manage my research, okay? I give them a protocol, it gets executed the way I ask them to, all right? We paired this with our on-farm trials protocol, which was a slimmed down, a little bit easier, a little bit more digestible protocol that we could execute on farm with private farmers who are not professional farm managers that manage research on a daily basis, all right? So we've been calling this a bit of a paired approach. So let's start with our protocol at our university research and education centers, 2 locations either side of the Chesapeake Bay. This is a pretty straight, simple nitrogen rate trial. Started with a 0, and ramped up, by, 30%. We are trying to hit that plateau. We're trying to over-fertilize some of this wheat and hit that yield plateau, all right? We chose to vary the rate that we apply, not at the green up application, but at the jointing application. We've seen some data from other parts of the country, some research that I've done, and I really think by varying the nitrogen that's applied later in the season, this is where we can have more impact, all right? I also think that this more aligns with our 4R and our nutrient management goals, right? Let's limit the amount of nitrogen that we're putting out up front that has a chance for loss. Let's focus that nitrogen at the end of the season when that plant actually needs it, right? It's like side dressing corn, all right? So we're already doing these applications, just somewhat shifting the rate that gets applied. So we did basically a straight rate trial at our research and education centers. I'm so sad that it doesn't appear that you can necessarily see the difference here. But because we were talking about field and plot layout, I wanted to give you a chance to see how I executed it on my farm. So let's just take one block, one rep here, all right? These first 5 plots are green. I know it's very hard to see that. And these are red down here, okay? So the green plots got 30 pounds of N in the fall, and the red plots got none. So all 5 of those treatments, half of them got no fall nitrogen, and half of them did. All right, we got 8 replicates here. All right, those were our rates again, and then we applied plant growth regulator on half. Now, I really would like our university farm managers to continue doing my research for me. So I could have made like each one of these reps, half of them get plant growth regulator, one not, and I could have randomized it. This was the best I could do to make sure that our farm managers didn't kill me and would continue to execute what I was asking them to do, okay? So that was my field layout. I followed pretty standard and solid field plot layout so we could run some statistics, all right? So let's look at the graph. We're going to look at the nitrogen that we applied, the total nitrogen throughout the whole growing season. We're just looking at wheat yield. I know Josh showed some graphs where we were looking at relative yield. We're just looking at 1 year's worth of data here. We're just going to look at straight yield, all right? So these are the different points. The 2 different locations are different colors. We plotted that nice quadratic plateau line, and I drew you visual lines so you could see where we maxed out on yield and at what nitrogen rate that was. And for those of you who can't read, that's fine. I need a reminder too. We maxed out at 97 bushels at 178 bushels per acre. Cool, all right? I'm not gonna go deep down this. I just wanted to show this. Again, like for thought and for interpretation. I also drew a line where at 120 pounds of nitrogen, which is basically our recommendation, high management wheat, you get 1 pound per bushel expected yield, plus an extra 20 pounds, so that's about 120. That was 91 bushels. Is it really worth 58 pounds for 6 more bushels? Again, open to interpretation. I'm just pointing that out there. Depends what lens, what glasses you're putting on, all right? So we also saw a 7 bushel increase with our PGR versus none, and with that 30 pounds versus no fall in. Again, 7 pounds worth it, or 7 bushels worth it for that extra trip, extra application, up to your interpretation. I'm just simply showing you my results at this point. All right, let's look at our findings at the on-farm. Oh, sweet, 15 minutes, we're rolling. All right, so as I mentioned, this was a bit of a paired protocol, paired trial. So I couldn't expect our private farmers who were so gracious to volunteer to do these trials with me to execute what I just asked the farm managers at my research center to do. So we really we had to somewhat slightly change our expectations. So this was the planned treatment structure. This is what I advertised online. Hey, come to my trials, are what my trials are, right? This is what I was going for. Treatment 1 is always farmer practice. Whatever you standard do in your field, that's what we're gonna do, all right? I was still interested in comparing 0 or 30 pounds of fall N, all right? I am still not varying what's being applied at green up, and I'm interested in looking at putting more of that nitrogen down at jointing, and then also a little sort of N rate trial. I wasn't going to ask these folks to do 0 N rate. Who knows? Maybe we will in the future, not the first year. So this is what I wanted to do at these 3 locations. We're going to go location by location, and you're going to see where we had to modify this a little bit, okay? So let's focus on our Montgomery County site. This one was the closest to what I actually intended to do. This is the only farmer who was willing to give me 0 fall in. So the other farmers were like, nah, I can't do it. I got to put out other fertility in the fall. We're just going to have to put down some fall in. So we pivoted a little bit. But in Montgomery County, we were able to basically execute the treatment structure as I planned. All right, I'm just showing you the highlights here. So I'm showing you the treatments, and I want to make the point that I sorted them in order based on the quantity of nitrogen that was applied at jointing. Okay, so the farmer practice, this was the lowest quantity of nitrogen at jointing. First number is what was applied at planting. This is how much N was applied at green up. Last number is what was applied at jointing. All right, so 40, 50, 50, 80, 80. That's what is really the most important thing that we're looking at here. And then that wheat yield, obviously. Okay, what do we see? So, very wide error bars. These 2 treatments were the only 2 that were significantly different, okay? Again, it's 1 year of data, okay, whatever. What is interesting to me, okay, next point I wanted to point out. These 2 treatments, while they differed in the amount of nitrogen that was applied at jointing, overall the same total N, all right? And you can see that there were differences in yield. What I'm seeing is the most interesting trend here. I don't want to spoil it because I want you guys to see it for yourself, but I'm excited, so I'm going to spoil it, all right? Look at the height of these error bars on this slide, and as we move through the rest of them. We're seeing decreased variability, shorter error bars, the more nitrogen that we're putting out at jointing. We're seeing less variability in that yield, the more we're pushing that nitrogen towards later in the season. That's, spoiler alert, that's the exciting thing that I'm seeing so far in 1 year's worth of data, all right? So that was Montgomery County. Let's take a look, oh, and then there was actually statistical differences when we compared those 3 nitrogen applied at joining. Clearly we're seeing higher yield, and again, shorter error bars, shorter variability. Cecil County, hopping across the pond over to the Upper Eastern Shore, all right? This farmer said, I can't give you a 0 fall in, Nicole. I got to put out 1 other fertility, even though I tried, get him some triple superphosphate, couldn't do it. So we slightly varied our fall nitrogen application, which is basically the same. And his farmer practice was he evenly applied the remainder of his nitrogen between his green up and jointing. So we kind of slowly pushed a little more and got a little bit of a end rate curve. So again, really important part of doing on-farm trials, you gotta be like a little flexible and you gotta be prepared to pivot and figure out what your end goal is, but pivot so you can actually execute it. All right, similar results here. We saw a significant difference in that wheat yield at that higher nitrogen applied at jointing. And again, I spoiled it for you. Height of those error bars, we're decreasing that variability. We're honing in on that yield as we're pushing more of that nitrogen being applied later in the season. All right, and finally our last site, Dorchester County. This is another one. It was just like, hey, Nicole, no go. We are not going to be able to put down no nitrogen in the fall. We got to put 30 pounds. So I'm not even sure what I was thinking. I probably should have just kept my treatment structure the same, but I was feeling saucy that day. So we ended up doing a little bit of a flip flop here. So treatment 2 and treatment 3 with the same total nitrogen rate. We just split which one of those applications got more of the nitrogen. Again, I probably should have just kept it simple, stupid, but you know, whatever. Feeling spicy, we decided to do this. And it was really to my detriment because we didn't really see anything exciting or significant, except the height of those error bars, decreasing that variability the later in the season we were putting that nitrogen. So again, I sorted these treatments here in order by the amount of nitrogen that was put out at the end of that joining application. All right. So 1 year of data, nothing too exciting. Oh yeah, that's right, my last graph. So then I just took all of my points and said, hey, what the heck? Let's just throw them on a graph and let's fit a line and let's just see. So this is the points at the research center, all the data points from the research center, as well as the data points from those on-farm trials. I'm approaching pretty quickly that blue graph I showed you that Josh got, with all those bajillion. Look, I'm on my way. I'm doing pretty good, all right? So when I plotted that quadratic plateau, we maxed out at 96 bushels at 180 pounds, which, as a reminder, when we plotted the same line for the research centers, we were pretty darn close, okay? Pretty close. So we're seeing data that is really aligning. All right, so after all that, my final plug, let me take a step. My final plug is I implore you, farmers in the room, and I implore you consultants in the room to maybe discuss with your farmers participating in these nitrogen rate trials, all right? We have eliminated some of the barriers to participation, okay? We are paying farmers, basically paying them to deal with me and Jean is what it is, all right? We know that it is a time suck to stop what you're doing, slow down, and to have to actually put these treatments out in the way that we're asking you to do. We realize it's a time suck. We're hoping that it's not going to be too much of a yield loss, but if anything, it's a time suck. So we're paying you to deal with us, okay? Giving you cash money to participate in these trials, okay? We found very quickly, after basically our first year of the on-farm trials, that there are folks who are very interested in participating in our trials, but they are limited by the equipment that they have, all right? So if you're talking with a grower, or you're a grower yourself and you're interested in participating, but your farmer's like, I really don't have any way to vary those rates or do the cool stuff that Nicole, well, have no fear. We have acquired some new equipment. We realized that this was a barrier for some people to participate. They just couldn't put the nitrogen rates out themselves. So we are in the process of getting this sprayer all set up. So we're still going to pay you to deal with me and Jean, but we're going to take less off your plate. We are going to come out and put out the rates for you. So if you're talking to growers who are interested, but they feel that they are limited by equipment, please still have them reach out to us because we may have the capacity to still let them participate without having to use their own equipment. All right. So I'm going to put that QR code back up here. This just takes you to my website. You can reach out to me. You can reach out to Jean. I've also put QR codes to 2 fact sheets here, talking a little about interpreting statistics and also interpreting variety trial results. I was a co-author on this fact sheet. We wrote it nationally. Me and agronomists, we put our head together and really we're looking at giving some very clear, clean guidance on interpreting statistics. And Andy Ness, who is the county ag agent up in Hartford County, he and I co-authored this fact sheet on the right about interpreting variety trial results. And if the QR code's not working for you for some reason, shoot me an email and I'm happy to share these files with you. So I just wanted to say thank you all for coming on this journey and talking about statistics with me, and would love to talk to you as consultants or any of your farmers about participating on these trials to help us collect some solid data to evaluate where our fertilizer recommendations are at for nitrogen, for wheat. So thank you guys.
Any questions for Nicole?
No. There's one in the chat asking if the results are from this summer's harvest or last.
The question that we have online is, are the wheat results from this past summer or the year before?
That was from this year. That was wheat that was harvested in spring, summer of 2024. Yes. The question was, are we pairing the trials with FSMT in the fall? Yes, we are. We are pulling FSMT samples. I have not looked at the data yet, but yes, we are. That's part of our plan, is pulling that as well. Good question. Yes, I saw you first. Sorry, I saw her first. We'll take as many as we can get. I'm somewhat looking for quantity, but also looking for spatial. We're really trying to get people across the whole state. I'd say 3 was manageable this year. We probably could take a couple more. So we would probably be looking for 5 or 6 farmers for this protocol this year. Yeah. So, and I don't think we have anyone signed up. I didn't check the Google form today, but we've really just started advertising. So this was kind of like the first event that we were really going to start pushing advertisements. So yeah, great question. Thank you.
When are you going to release the corn nitrogen?
Yep. The question was about when are we going to release or put out some of our corn nitrogen results. Hopefully soon. Technically this is our last year of the study, but we're going to extend 1 year. We were a little bit delayed with our execution with COVID. Like we got the money right around COVID, but we're starting to put our data together and really starting to crunch out some of those publications here soon. Yeah. Thank you. Thanks for the question. Gosh.
Said that they were applying about 30 pounds of nitrogen in the fall. About 80 pounds of phosphate out.
Yeah, it was because they were trying to put that phosphorus out with dapamap, and I offered. I said, "Well, we could get you some just straight phosphorus with that." I said, "I'll put that in as part of the trial," and I got a wishy-washy. I'm not questioning what their soil test is calling for at this point. What'd you say? That's a lot. We're good with just nitrogen right now. Thanks. Oh, sorry, I didn't repeat the question for those online. The question was about the on-farm participants. Were they using NAPRDAP to put their other fertility out in the fall? And I said yes, and I offered them some straight phosphorus, but it was not received with a warm welcome. So we let the farmers do what they were doing.
Thank you, Nicole. Give it up for Nicole. All right, so we have a good bit of left. Take stuff. If you know a family in need or if your church could use it, take it. I want it to be eaten. I want it to be consumed. So happy to pass it along to anyone in need. In addition to that, thank you again to our event sponsors. We truly appreciate your support of the 4R Alliance. A few housekeeping notes. All of the credit links should be in the chat box for those that are online. Where lunch was served, you'll be able to find the credit sheets for all categories. So on your way out, stop and sign the appropriate piece of paper, and they will not be submitted till Monday morning. So do not email me tomorrow and say my credits have not been submitted. I am going to Texas tomorrow, so I will apologize for that delay now. We appreciate your time coming to our second annual 4R Symposium. For those that are interested in the Delaware, Maryland 4R Partner Meeting, that is scheduled for October 28th at the Y Research and Education Center. You can scan the code on the 4R table to register. And then we will also be sending out a follow-up wrap-up. So please utilize that email to give us some feedback for our future events. We love to hear who you guys want to hear from, what worked, what didn't work, and your feedback. So once again, thank you all for attending today in person and for online. We appreciate your support and have a good, safe drive home.